one of them who was in China as a teacher.
And here we are.
Oh, there we go.
So we just got a kind of a bad connection before.
Mm-hmm.
So we're now streaming live on Facebook, and I'm going to guess people might be popping in throughout the afternoon. I'm Ted from a company called Magnitude, and we're working with the National Laboratory and some cool folks all around the world over the next 30 or 40 days to introduce some really fun, exciting ideas that students don't necessarily even need to have a computer. So if you're an educator or a parent, you can pick up what we're sharing with the other folks that jump in and share with us what's going on, and you can bring it back to your students anywhere from pre-K, honestly, even all the way up to the university level, as we'll be talking to some researchers doing extraordinary things on station. In fact, we're kicking off here on the first day of spring, so happy equinox, everyone, with Dr. Richard Barker from the Gilroy Lab. And the Gilroy Lab has done extraordinary work in plant biology, in microbiology, and really astrobiology as a whole. They have an amazing data set they've collected from all types of research around the world, on the International Space Station, probably on the shuttle as well. And Dr. Barker's here. Thank you so much. And I saw your dog Savannah's joined us as well, so we might see her a little bit more as well. Dr. Barker, thank you for joining us. In these times of uncertainty, we hope, we're basically every public school is now a home school. Maybe you can share some insights and some amazing stories about what you guys are doing in the lab and how that might directly affect communities around the world. Maybe just a little background, I think, would be a good place to start as we kind of get warmed up here.
Okay, so if I could introduce myself. Oh, I've got a video for this. Should I use the video?
Sure.
Okay, so
do we get the cool music?
I'm gonna share my screen if I may. And then I'm gonna go to, and anyone can go to this website. It's astrobiology.botany.wisc.edu. So this is, okay, this might be a bit bigger. This is the Gilroy Life Science Lab. So I get to work with Professor Simon Gilroy. He looks like Gandalf. He's absolutely hilarious. And our mission is to promote collaboration within the life science community, particularly with the goal of taking what we learn about life science on Earth and applying it to extraterrestrial environments. Can you see this website? Is that?
Yep.
Okay, cool. So initially we're Plant Science Lab, but now we get to have fun in outer space as well. So we just want to use kind of extraterrestrial environments.
Right.
There's an echo coming.
Yeah. If anyone's running the Facebook, just make sure you've got your mute on. That was me, I think.
So we're interested in plants. They're really sensitive things, and so we think that they make the perfect canary in the coal mine. So in the olden days, when miners would go down underground, they'd take canaries with them, and if the canaries passed out, it could be caused by a gas leak, and so they see the canaries unhealthy, and so they get out, and the explorers, the miners, are then saved from the hazard. So by putting plants into certain potentially dangerous or alien environments, we can learn a lot about them and perhaps get some kind of warnings about how life would adapt to these strange extraterrestrial stresses. So, but in order to do that, you have to kind of believe that plants are sensitive. And so we've been looking at plant sensitivity for some time. So I'm going to play this video and see if it goes full screen. The music is off. So I'm going to cut the leaf off just there. Did you see that in the bottom right-hand corner? And now an electrical wave is moving through the plant, this neighboring leaf, and is activating the plant's defense system. So this is quite an extreme example because we're decapitating a whole leaf. So this would be like an insect eating a plant in your garden. But the stresses that we encounter in spaceflight are much more subtle. Things associated with microgravity, things about cosmic radiation flying through the organism. So we've been trying to investigate this. And the more we've learned about the plant system, the more we realize it's similar to mammalian systems. So this is the same plant, and we're adding a chemical called glutamate. Glutamate is an essential part of your nervous system, of all life's nervous system. And we're just adding it to the plant, we're showing the whole system is entirely connected. So what happens when this sensitive being encounters microgravity? This system has always functioned with 1G acting on it. that mechanical force that's always there. But what happens when that's gone? We know from astronauts that our bones start to deteriorate, our muscles start to waste, and similar things start to happen in plants. And that's what got us really excited about using the spaceflight environment as a way to investigate this system. So we were terrestrial biologists first, who then realized that the extraterrestrial environment provides a really novel way to investigate phenomenon that we take for granted here on Earth. And that led to 3 successful space launches. And the Big Ten network made this
Humanity is
video.
to become a multi-planetary species and have people living and working on the moon and on Mars. We need to ensure that we can grow food locally for them. We've had 3 successful space launches, and we're the first lab to say that the plants that we engineered for the spaceflight environment grew better than the wild type.
Space is a stressful environment because of the lack of gravity. The Gilroy Lab studies how plants grow in space to better understand what it might take to have strong growth and good yields to support astronaut health. A lot of students at the University of Wisconsin have been involved in helping us explore the research that we're doing with NASA.
We need to work with engineers and computer programmers to develop new tools in order to create stressors on Earth that replicate the spaceflight environment. This work can be used as a roadmap in the future to accelerate the adaptation of plants to the spaceflight environment.
So
Fabulous.
as a result of these spaceflight studies, we've accumulated a load of genetic data about how plants respond to spaceflight. And this gave us the opportunity to start to work with a program called NASA Gene Lab. NASA Gene Lab is a collaborative organization within NASA, which has collected together all the life science data that humanity has generated using spaceflight as a novel stress. And we've been creating visualization portals to make these data available. We've got a couple of videos that have been made to kind of promote our work, but for me, it's the subtle things, it's the simple things that happen now and then that you take for granted that makes spaceflight research so much fun. So for example, on our last spaceflight study, we had an astronaut harvest these samples for us. In the day-to-day running of a lab in a research classroom or whatever, one of the things we regularly do is open a Petri dish, take plants off that Petri dish, and then put them into fixatives so we can explore their genetics by doing DNA extractions and those types of things. Usually when we do it here on Earth, we have gravity acting the whole time. But here in this video that I was just playing of the astronaut doing it, you can see there's these certain moments when gravity's not present and you just see the camera floating beside him or the Petri dish floating in front of him. And so it kind of takes those day-to-day things that we have to do life science research here on Earth, and it adds this really kind of alien component, which is, it's so subtle, you know? But so through the excitement, through these novelties, we are still doing some really interesting biology and trying to make these data available to people. So our first goal is plants, and our secondary focus is microbes. You can't have plants without microbes. And so we have to appreciate until 5, 6, 7 years ago, everybody in NASA thought microbes were bad. Any microbe is a bad microbe, kill it. It might be poisonous, it might be a pathogen. But nowadays in the microbiome era, post sequencing, essentially we've started to realize the microbiomes are natural and we need to monitor them and try and nurture them. And so I just want to play this video here. This is talking about the built environment. This is I've borrowed this from the National Health Institute of Health, and this is a water molecule and a droplet even, and you can see microbes living in that. So this is like a graphical representation, which is very artistic in nature, which is designed to make you look around the world around you and realize that microbes are there. And not in the way that the current COVID-19 pandemic is making you look around and think, oh, microbes are there, be afraid. Instead, the goal of this is to make you think, wow, microbes are inside me. There's more cells inside me from microbes than there are that are human. And so when humans go to outer space, when we go to the moon and start mining there, and when we go on to explore Mars, we're going to be taking microbes with us. And they've always been there and they will always be there. And so we need to appreciate them and understand them more thoroughly, because if we don't take care of them, they do have the capacity to turn pathogenic. And communities can start off as like a nice, healthy system full of symbiotic relationships. But again, if not nurtured in the right way, they do have the capacity to turn pathogenic. And so this is why we want to keep an eye on the plant on the microbial population of the space station, with the goal of informing plant scientists as to how best to grow plants in this alien environment, with the concept, the knowledge that plant pathogens are there. And so we're shortly after this, I'll show you how to access these data and how to fish around and extract things that might be of interest to you, because there's thousands and thousands of microbes on hundreds of locations. So
and Dr. Barker, you actually kind of created, in essence, a census, if you will, right? Of like who's up there.
Exactly that, yeah. It's a list on a bunch of different locations, and so you can see where those microbes are too. And so we'll come back to that in one moment. Last thing on this page before moving on, at the bottom here we have a couple of web links. We have a sister website called astrobotany.com, where one of my former students has been making educational introductory material, so that anyone can go visit that website. He's also got some cool t-shirts that I like to wear.
I like the toaster.
The toast. Oh, that's the best thing about when you get your first launch with NASA. You get your mission patch.
Exactly.
So we've gone through toast 1, 2, 3, and that's actually a great segue. Toast stands for Test of Arabidopsis Space Transcriptome. So we mentioned earlier those first flights that we did. We put plants up there. We monitored the gene expression pattern for every single loci in the genome of Arabidopsis. And that data is now on this website in a way which requires no coding and allows you to compare that to all the other previous studies that were done. And I'm doing my best to keep it up to date, keep adding more. If you want to get a feel, because I'm just diving straight into the high end like research genetics, because that's what we do with NASA and GeneLab. But like, if you want to get like the background for more of an easy read for a younger audience, we have got this peer-reviewed publication. It's this button just here, in which we just kind of quickly go over in like a couple of pages, like the broad spaceflight, like astrobotany research area. So these two links down here, great introductory material. I recommend it to anyone who's new to astrobotany or astrobiology in general. Okay, cool.
Dr. Barker, if I can, because clearly you're right at the very edge of research and discovery, understanding how plants behave and the different kind of activators. And we have some educators that are joining us that might be working with much younger students. So I think that's the role when we kind of play over here at Magnitude, where we have kind of the macroscopic investigations, and you're doing these microscopic investigations. And so I just want to remind everyone that really why we're bringing this collaboratory is for finding things for your students to do. Dr. Barker and a collection of scientists and engineers that are working directly with the International Space Station. The space station is an amazing invention, creation across 18 different countries. We'll talk a little bit more about the International Space Station throughout the afternoon, but I just want to make sure you understand that this context of what we're creating is something for your youngest students. Maybe you're a parent thinking about something for your child to do, or maybe you're an older student that just dialed in and because you're interested in what we're talking about. And there's a true opportunity butt right against the research as it's happening. So please, Doctor, go on. I just wanted to make sure folks knew what that was about.
That's excellent. I appreciate the tennis. That definitely helps. So yeah, I just logged into astrobotany.com. So as you can see, there's a few resources along the top here. And we used to have a Astrobotany. So this is for the youngest introductory audience, Astrobotany TV. It was on YouTube.
Awesome.
And again, so this was one of my former students, long-term friends now. You know how life is. They grow up. There's a series of interviews and some videos that he made in the lab. It started off as a bit of fun, but there's a bit of a theme to it.
Guys, I've got some bad news for you today. This may come as a disappointment to some of you. You may notice that I'm missing my chain.
So there's some jokes that are aimed at a different audience than me, but
there you go. You're talking about like sequencing or what have you?
Yeah. So check out Astrobotany TV. It's really fun, and if anyone wants to continue developing this, we've still got some of the resources here. So So I think it's really important to use the arts to tell the stories, because when we just start diving into the microbiomes and such, sometimes the message gets lost. So life science laboratory, the story. Okay, cool. Microbes or plants first? Let's go for microbes.
Let's start small. And
Yeah.
while you do that, we have a guest that's actually logged in. I don't know what time it is. I'm going to guess it's in the middle of the morning from Nigeria. So Olayinka, welcome. Thank you for joining us. We've got some countries in Africa that are starting to run our experiments in space.
Yeah, hello, hi everyone. It's not middle of the night. It's actually 21 minutes past 9 p.m.
Oh, okay, so not bad.
Yeah. It's a pleasure to join you guys.
Yeah, Olenka is in charge. She is the Assistant Chief Scientific Officer for the National Space Research and Development Agency in Nigeria. So, who have launched a number of satellites, and I've worked with Olenka over lots of outreach across the country. Anyhow, thanks for coming on, Olenka.
Yeah, you're welcome.
Yeah.
It's a pleasure to be here. Thank you.
Yeah. And I'm sure we'll get back to you soon.
Okay, great.
Okay, great.
Wonderful. In fact, just a footnote there, we're going to create a separate conference for our friends on the African continent, probably as early as next week. So we'll keep you posted on that. Would love you to join us on that one. So this, you shared with me the other day, Dr. Parker, is absolutely phenomenal. Please go on. This is cool.
Well, this really is the beauty of the International Space Station. It really is a great place where lots of people have come together, and humanity has created something which is truly awesome. It's a completely unique laboratory. It's been up there for about 20 years, about the size of an American football stadium or an English soccer stadium. But people have been living and working up here continuously for 20 years. That means microbial communities have been here up continuously for the last 20 years. And so the International Space Station has some really interesting selection pressures associated with it. The increased cosmic background radiation is one major factor, as is the lack of convective mixing of gases due to the lack of gravity. Because essentially, although this 3D model is just like static, it's falling. It's falling all the time. And that creates a sense of microgravity. So that stops buoyancy moving gases and liquids around. So if you're an astronaut and you go to sleep in this system, when you sleep, you breathe out plumes of CO2 as you breathe in oxygen. And so essentially, without convective mixing of gases, this astronaut who's falling asleep here is creating a big bubble of CO2 around his head. So we need fans to circulate the air. So life is encountering localized hypoxia. And you can see footprints of that in all the genetics from all the studies in orbit. It's pretty much a generic theme. What we're seeing is slight changes in the mitochondrial function, changes in the TCA cycle. So if you live up in microgravity for a long time, you get used to having a different amount of CO2 and oxygen in the air around you, just like when humans go on airplanes. So every time you've gone on an airplane, you've got somewhere, maybe you've got groggy and been a bit sleepier than you thought you should be based on how long you've been there. And that's essentially because of the change in ratio of CO2 and oxygen during that air flight. And the same is the major selection pressure on the space station. The others is that the intergalactic cosmic background radiation. So we're going to be examining some of the microbial populations on the station shortly. So here we'll look at the dining room table, we'll point that out. I'm also going to point out the advanced exercise resistance device. Yes, the bicycle thingy. And we'll look at the microbes that live on there in a couple of moments. But I just want to have a quick peek at radiation before, because I want to think about the selection pressure that radiation is.
I just want to jump in here. Excuse me. That is just the most amazing animation tool that you used.
Isn't it?
That was extraordinary. Wow.
I think that's East that did that. Isn't that right, Doctor?
Oh, we haven't even gone on the station yet. No, this is from another organization, but we're gonna fly through the station to go to the exercise resistance device and the dining room table. But I wanted to look back at the radiation story one last time before going to that location. But so the space station is flying around the Earth, and the Earth is being protected from the sun by its magnetic sphere. So this is the Van Allen radiation belt. You can see it kind of throbs annually. So if you're living up there, oh dear, sorry, my bad. If you're living up there, where is it? 2 seconds up there. Yeah, then you're getting more and less radiation coming through you progressively in like waves coming back and forth as the Van Allen belts throb. There's one other interesting concept to note, another way to look at that, and this is courtesy of ESA. So this is the Earth. We have the magnetosphere around us. We have those Van Allen belts. And there's a point where they come low to the Earth. This is what we call the South Atlantic anomaly. And so this is due to something physical associated with the Earth's magnetic sphere that is beyond me. But now you can see that white line, that's the space station flying through it. So if you look at the data for the space station's radiation, you see this like, how long does it take? Was it 4 hours? As the space station goes around the Earth, you see that oscillation of radiation. I forget how long the day of an astronaut is now.
Well, it's a 92-minute revolution.
That's the one. That's the one. Sorry. Yeah, so you see that 92-minute oscillation as they go through the South Atlantic anomaly, depending on the sphere. But so there's more of this cosmic background radiation. And how do we think of this radiation? Well, we have the charged hydrogen ions that are being blown off the sun. So this is your like solar wind that's kind of floating around. But then they're the biggies. And these are your intergalactic cosmic rays. And so we can do the linear accelerator. And so we can do our best to simulate intergalactic cosmic rays using linear accelerators. And essentially what the linear accelerator is trying to do is be in a supernova. So in a distant solar system elsewhere in the Milky Way, a sun has gone supernova, it's exploded, and it's fired out a load of charged ions. They've gone so fast, they've lost all the electrons, and so you just got these charged nuclei of like oxygen and gold and iron just flying through space. And so this is this thing charging up. This is the best humans can do to try and like simulate that. And so there's not just one ion, there's a collection of different ions that get fired. And these are the things that are fascinating because we get exposed to them on a low level on Earth, but the Earth is protecting us from like over half of the universe. But when you go away from the Earth, when you go to say low Earth orbit, or when you go even further to like cislunar orbit or all the way to Mars, this is when you start to get hit by these massive like charged particles that are just going to come flying at you. Okay, so we've got cosmic rays. It's kind of fun, isn't it? So what happens when you live up there with cosmic rays for say 20, however many years that is, 20 years. Just trying to, this thing, if I can just zoom out a bit. Life has always had radiation, right? Like this is real-time monitoring of the Earth's radiation thanks to URAD. And so you can see radiation is prevalent in many of the places that humans live all the time at a rate that can be measured and is constant. So what happens if you then have that extra level of that intergalactic cosmic background radiation as a selection pressure? Well, let's have a look. So we're going to think from the community level first. That's why microbes are great. But we can also think of it from the individual species perspective, i.e. what would a plant feel? How would a plant feel about that? Or what would a mouse feel about that? Which is the major model organism for research into the data associated with like biology's response to radiation. It's easy to put a mouse in front of a particle accelerator. It's less easy to get permission to put humans. So mice have become the predominant model. So this is all coming from NASA Gene Lab. So before delving into that, I just want to quick shout out to NASA Gene Lab. So this is the organization where we get all this data from. So they've been collecting a repository of data for the last 5-6 years and building like a collaborative workspace and trying to make it so people can access all this other life science data. So, this is a new one I haven't seen before. Swabs from the International Space Station raw sequence reads. Okay, sorry, there's always new data on him, and my mission is to try and keep on top of it. So this is
get a neural chip when they're available, Doctor.
Yeah, right. They're just constantly dropping. There's always something new. So again, this is like the microbiome of plants that are going on the space station. That's a paper that's just come out. All the raw data is here. So big shout out to NASA Gene. I've been using their raw data and then using some of their processing systems, occasionally using my own to make the data easy and accessible. What do I mean by that? So what is raw data these days? Okay, so in the post-genomics era, I like sequencing DNA and RNA. I like to see which genes go up and go down. If I'm looking at an organism, I can measure like one or two things, like the size of its nose, or like how fast it moves around. One or two things I can get from photos. If I get a sample of that organism's like DNA and RNA, I can monitor, say, 25,000 genes all at once. The data is much bigger, it's much richer. And so we can start to get more insights into how organisms are responding by looking at the genetic data. Ideally, in the perfect world, you link the genetic data to the pictorial data, the physiology data, and the two things tell the whole story. So far at GeneLab, NASA, they've been looking at the genetic data in isolation, and I'm kind of pointing out an opportunity for the future here. And we've been pulling patterns from that genetic data to make predictions. And what I now want to do is grow the plants, grow the organisms, and then see if we can observe any of those patterns that were predicted by the spaceflight genetic data. If only we had access to opportunities to design experiments to fly and students that wanted to explore these data and find these patterns and then design those experiments. Anyway, food for thought. Okay. So before coming back to the plant genetics, because you can see that's my real passion area, plant physiology and plant genetics. Again, we really got to understand the environment first. You know, I'm really an environmentalist at heart. I really am. When I went to college, I just wanted to do environmental science to save the planet. But then, but then I started realizing that, well, plants are the crucial crux of this like food chain, which keeps humanity alive, which allows civilization to exist, and allowing certain people to do amazing things like make a space station. And so I realized, like, actually, if we can fortify that the food production and help that be more sustainable, that would be a better way to ensure that we reduce our impact on the natural ecosystems around us. So by focusing on intensification of agriculture, we can defend the ecosystems around agriculture, because if we don't, it's going to spread. So this kind of interest kind of brought me to doing this kind of spaceflight research. There's a loop that I was about to come back around to. So understanding the plants was so important to ensuring we have this foundation. But in order to understand the plants, we have to think of them in their environment. And that's when I became fascinated by microbes. Initially, before that, I just loved yeast because of the its role in brewing and many other baking traditions. But I realized there's a lot of other really fascinating microbes out there, but there's so many. Oh, there's so many. And over the years, people have done such a good job like classifying them and reclassifying them. The classic phylogenetics was confusing to me. And so what I did was I did what I tend to do when something confuses me, is I made a new friend and I asked for help. And so
that's my rule book.
Right. Yeah, they don't teach that at school, but they should. So
Save me, Mr. Wizard.
It's true. Like, often you're told that you have to pass the test by yourself. It's not the case. You just have to be polite and humble and work out what is the right question and who is the right person to ask. And so eventually I was able to connect with the head of the Interplanetary Protection Team, Professor Venkat Swarupham from the NASA Jet Propulsion Laboratory in Pasadena. And him and his right-hand man, Nitin Singer, were really eager to help and collaborate. And so what they've done is provided the microbiome of the International Space Station. And I'm just going to play this little video, and then kind of talk over the top, if that's okay, because it's got my terrible electro music that's playing in the background. So
I kind of like that.
Oh, I'm glad you did. My mum didn't, so that's why I keep playing it, downplaying it. So when we go to Mars, we're going to take microbes with us. I played this video earlier. I think that's fascinating. And well, these little things popping up, these are chronoplots. It's just an interactive data visualization that allows you to explore the microbiome of an organism. And then what we're going to do, we're going to take you in, just showing the user how to find mango. It's on astrobiology.com. Astrobiology website.
Yep,
So
I posted it in our Facebook feed, and I'll post Mango direct link.
Perfect. Appreciate it. And so in there, I've hidden like the original paper. So the original paper published by my friend Nitin and Venkat is there, so you can easily quickly find like the background literature that's been published on this study. And then what we're going to done is made an alternative way to explore it. I'll show you this thing in a bit. This is a gift from ESA. So this is a mechanism that allows you to fly around the space station and look at different locations. I'm going to fly all the way over to node 1, because node 1 is where the dining room table is. And that's my favorite point of reference. The reason I like the dining room table is because I know what that looks like in my home. So it gives me a point of reference on the station. A lot of the station is a bit weird and old, so it's got funny names. And then on the right here, what you see, this is a pairwise comparison. It's what we call a scatter plot. And we can select 2 different locations. So I'm selecting the dining room table and the advanced exercise resistance device. And we're now looking at all the microbes that are on those 2 locations. And we can see the ones that are most abundant, like on that diagram kind of jumping out. And then along here, we have all these different filters that we can apply. And so we can start to search for different microbes that have different characteristics, such as the capacity to form biofilms, capacity to have antimicrobial activity, to be human pathogens, animal pathogens, plant pathogens. And then there's these other dashboards. So these ones now allow us to look at the different locations. So this particular one is looking at the toilets, the waste hygiene compartment. In the top left-hand corner, you can see there's a couple of filters that have been applied. So we're looking at a subset of the microbial population at that location. And in the bottom here, we have the PMA and non-PMA treated. So if it's PMA treated, it's only living samples. PMA is a chemical that binds to the membranes of the organism. So then that way it keeps it intact. So you can select for the microbes that were alive during the sampling. So we've got both like a rough overall count based on all dead and alive DNA samples. And we've also got a count based on alive living samples. And so in this particular example, I'm just kind of poking around and looking at the my favorite particular microbial community. I've hooked up to this database, the pathogen interaction host database. And so you can search based on the sort of disease that you might be interested in. And then
you had a favorite, but you didn't tell us what that was.
So
my eyes are kind of old. It's hard to see the little fine tuck there.
Yeah. So we have a theory that about a certain variety that might be radiation resistant. Especially this background like pressure, selection pressure that could be organizing, that could be on them. And so this then leads us to a collaboration that so that's the space station microbiome, and we found this microbe that's up there and it's in lots of these places and maybe it is radiation resistant. But how do we know? There's no ground control. Like there's no space station on Earth, right? What is the ground control? So I started a collaboration. So again, I asked for help. So I started a collaboration with the European Bioinformatics Institute, a lovely group of people. They have this thing called Magnifi. And we pulled in the 10 studies from NASA GMAB. So there's 10 microbiomes in here. They've now all been analyzed and anyone can go in and access the data. The hard part is writing the story afterwards. And if anyone wants to do that, please reach out. We can collaborate on that. But what we then did was, so they processed the data in here and then compared it to the Earth Microbiome Project. There are these 2 big, really huge microbiome studies that have been done in recent history. One of them called the Earth Microbiome Project, the other one called Tara Oceans, as they did all the oceans. And there's hundreds of thousands of samples in there. And so when we looked into these 2 massive databases, only 160 out of the tens of thousands, I don't know how many thousands there are, only 160 had this microbe that I think is radiation resistant and which I think is being selected for on the space station. So we're now searching for more data because it's, as a scientist, you never have the luxury of being confident. You always have to be trying to search for another way of proving or of finding more evidence to support your hypothesis. There's this scientific reasoning which is really important here. So, that was why I asked for their help. So we could try and people come at the same question from a different perspective, to producing different data from a different perspective, which may or may not corroborate our theories, our findings. And this is
triangulating, kind of triangulating your treasure hunt here, aren't you?
Yeah, and that's fine. That's a lot of people think like science is this, it's just right or wrong. It's it's not a religion. It's not like some sanctity law, which is sanctity law that predates us. It's like it's this living, breathing, growing concept, this collective consciousness of data and knowledge, and things have been tested again and again, have been proven. Like it's not a flat Earth. So sometimes in this post-truth era, we just have to we
should have started off at the top with that one, right? It's right there behind me, live from the space station, live feed from ISS above, which is kind of cool. This inspires me. If I get a little depressed, I just turn around and look at the Earth from 250 miles up.
Mm-hmm. It's amazing when you look at the world from outer space. You just get completely different perspective.
This map sure looks flat to me, though.
Yeah,
that's right.
All right. How do we mute Liam?
It's called a flat panel display, isn't it?
The space station falls off the flat Earth, at least once in orbit, and then somehow magically comes around the other side. Wow. Anyhow,
I love the foundation you're building us before us, Doctor. It's just like this foundational thinking in the site. If the big takeaway for me, if everyone kind of jumped in, it's like, oh, this is really deep, right? And it is. It's fascinating. Is that when you're excited about something, but you're uncertain about the future, is go find some help. Go find others that really kind of dived in and know.
Yeah. Exactly that. And then sometimes kind of reset and go, okay, what is the bigger, broader question? And so before diving into the first database, I'm just going to play. Actually, no, we're going to dive into the first database, and then I'm going to play you this video that'll take us back to plants. Is that okay? Yeah, let's do that. Okay, so. Oh, great. Obviously, it's logged out and logged in again. So I build lots of these data visualization systems where I've been amalgamating these data together. So this bit of a, okay, this is the plant genetics one, TOAST 4.6. So we'll come back to this one later. So each one of these icons represents a different spaceflight study that's been flown. And they've all been connected together to make a genetics exploration system for students to use. Initially, it was designed for professors, and then we made the system so easy to use. We tested it in high school, and the high school kids were able to find the same patterns that we were. So it was really pleasing.
Can I ask, is this available to the public? I see it sort of like an IP address up there for the web server. So is that internal or is that accessible external?
That's a great question. You're all right. This is my internal window into it, but it is totally available on the Rights website. So this one here, Astrobody Toast.
Okay.
This is that's the plant genetics. The one over here, the Astrobiology Gene Lab. This then goes into Drosophila, nematodes, all the other model organisms, humans, mice. It's much broader and bigger, but it's not plant focused. And the next one is
Dr. Barker. So I've got this just extemporaneous idea here. Let's create like a high school treasure hunt in your databases. We take this offline, or maybe we just riff on it, but wouldn't it be cool if there's an area that you really know, maybe you know this bit of the forest a little better, and we can challenge our high school students that are popping in, or even middle school, if you really want to get ambitious. Say, here's a quest we're going to send you on, because we have some of the answers, we don't have all the answers. And if there's an easy way for the public to access Toast, well, let's bring them in. And as long as it doesn't create too much of a bottleneck of bandwidth for your research, I'd love to kind of pursue what we might be able to do with that and share it with others.
I mean, that sounds great. I think I forwarded to you in our last Zoom meeting the publication, the one that came out like a week ago, last week, in which we only did like 3 or 4 mini comparisons within the tool. The tool can do thousands of different comparisons. But in that paper, we only need to do 4 comparisons to find a really good story. And so we haven't done all the other comparisons. And this is why we need the help of the students. This is why we need them to go in, explore the data, go through it with teachers, ask questions, and then hopefully generate hypotheses and to write more papers, because the space station is the most expensive thing humanity has ever built. It's awesome. It's a completely unique research environment. There's so much we can learn, and we only have it for such a short period of time. This is why we made the tool, to put the data into as many students' hands as possible, so they can help us ask the next questions. Because you're going to be flying stuff, we're going to be flying stuff over the next few years, and we want to make sure that the questions that we're addressing are valid and are useful based on the previous studies that were done before. That's why we've been doing this for Gene Lab. This is why I'm so excited to be here with you today.
Yeah, we're excited you're here as well, that's for sure.
So before delving into the plant genetics, this is the Microbiome Explorer. And should I just take you into a typical way I go in?
Oh yeah.
Usually, so these are a bunch of different apps. So we can look at the database in the phylogenetic tree, like kingdom, domain, phylogeny, class, family, genus, species. I like species the most out of that lot. We can look at the actual phylogenetic tree and the dendrogram and through networks and that kind of thing, but that stuff's not so useful. So the first thing I tend to do is think about it from a location-based perspective, because I'm thinking about where's my plant going to be and what microbes are going to be near that plant, because that's my personal perspective. But hopefully other people, when they go into it, they may have a different question, a different idea. So I'm just going to take you along my little tangent. So first things first, I want to go to the space station. I want to fly around. I want to experience it. Like, some people are astronauts, some people are astronomers, some people are astrologers, you know, that's fine. But we can all be a little bit of an each. We can all be a little bit of each. And I think that's important. So this is that app that was made by the European Space Agency. So I'm just going to kind of like fly around, go to node 2. Like, so maybe you know the layout of the space station. If not, hey, go for it. That's a fun game in its own right. There's ISS Plants is a I think it's called ISS Plants. There's another game. There's an app you can get where you can fly around the space station and learn your way around it and eventually find the hidden veggie growth system. But the thing I'm going to search for now is, this is the US lab. That's cool. This is where one of our first experiments, just down here, it was like plugged in somewhere. Where's it gone? It was just a brick, so like it didn't really need anything. Like, so early experiments are nice and simple, things like bricks, just biological work, research in a canister, it's just a petri dish and dark. But some people actually live up there, not just doing experiments. And this is what, when you live somewhere, you need to eat. So this is the dining room table. You can see it just there next to all these awesome mission patches. Okay, so we're in node number 1. Okay, so it's good to know.
Yes, and so if you do have a question, by all means, post a question in the Facebook chat, and we'll, if Dr. Barker has time with us, he'll answer your question if he can, or really anything related to what we're doing in space.
Exactly that. So yeah, and then we're back to this menu again. So I mentioned earlier that I'm fascinated by dining room tables. I'm just looking at the names here. Copula. Oh, that's the window. That's a pretty cool place. Oh, but I want to go to dining room. Oh, there it is. So we go to the dining room table. But again, like, the copula is one of the coolest places on the space station, right? You can go there and you can look out at the planet. That's where all the astronauts have their selfies taken. Okay, cool. So now I've got another dashboard. I don't like data. Who are you, crazy people? I love data. Okay, cool. So we have these things here. This is telling us the microbial population that's been measured here on the dining room table. You can see in these colors here, we've got 3 time points: March, May, March. So this 2015, 2016, and in the near future, we'll be getting more data from them. This was just the first one that's out. So this will grow and get hideous probably. Okay, so this is like going through time from left to right. And this thing here below, this is the quantity of the microbes going up here. And there's so many that's being listed, like it can't even write the names out in full. There's no point. Yuck. Okay, so we can filter. Like, maybe we should look at, instead of looking at all the microbes all at once, maybe if we have a hypothesis, we have a conversation with a friend, we have an idea about a reason for selecting a small proportion of them, then we might see a pattern in that small proportion. So as a plant scientist who wants to grow plants on the space station to eat, I want to know, are there any plant pathogens that might harm my plants? So I'm clicking on plant pathogens, I click on yes, 54321. So, okay, this table here, you can see in the corner, this has got all of the plant pathogens from the plant host interaction database listed. So now we've just gone and put a plant pathogen filter on and selected a subset of the microbes. You saw a bunch of these lines all disappeared, and now we're down to just a few. And look at this one. What's that? Ooh, these are my favorites. Okay, better not show you that one. But okay, you can kind of see the game now, right? So maybe you are not a plantophile like me. Maybe you prefer humans and animals and such more. And like, or maybe you're interested in human health. These pathogens are of interest to people who are interested in clinical biology. Who would be a health worker? Anyway, so you can see there's just a few of these different tools. We go to the species level. So another way to do it. So there we were selecting based on like the filters along the top. Maybe you're interested in a particular type of microorganism. Maybe you've got a favorite model species or like genus or something that you're interested in. So maybe I'm going to search for, so just down the bottom here, I can just type in meth. There we go. All those methylos, because I'm interested in methylo stuff, aren't I? There we go. So you can see, you can approach the data from a range of different ways. And so it comes through the conversation. So perhaps you have a grandfather who had some illness and some bacterial infection. You could search for bacterial infection. I think Legionnaire's disease is up there. All the graphs are supposedly interactive. So you can kind of just come in, click away on them, and then that will create a subset. So now what we've gone and done, I've gone and selected the top 7 most abundant species that were found on the resistance exercise device on the node 3 on March 4th.
You can probably identify the astronaut, I guess, right? Through a little forensics.
Well, it's funny you should say that. In the first version of this, yeah, there was human was the major. We actually had to remove all of that for
That makes sense.
That, that like security thing they make you do. But yeah, you're totally right.
The data.
There was also gorilla up there. I think that was a mistake in the code, and they just wrongly assigned some of the human DNA. But
well, there was a gorilla that was launched for Scott Kelly. It was a joke by his brother. He sent up an outfit, and then for his birthday, Scott Kelly dressed up in that.
I wonder if he rubbed himself all over.
Anyway,
rubbed it all over
so I wonder whether that was so maybe it's actually when he was wearing that. I don't know.
Well, it's funny you should say that, because I wondered that same thing, because I thought, well, first of all, it's a gorilla suit, so surely it's all polyester. But okay, hear me out. If you've got a gorilla suit, what's the first thing you do? You go to the zoo and you try and hug a gorilla. So maybe that gorilla suit was used in a pre-used.
So his expedition 44, 45, 46, that was in 2015. So if this data's back from 2015.
I think it's close.
Oh,
It's close. There you go.
Oh my gosh. Scooping it out. He rolled around in the gorilla den before he shipped the suit up, is what happened.
Anyhow, but back to you.
That's wonderful.
That's awesome. Okay, cool. So we've done some ecology. We've been fishing through, looking at subpopulations. You can think of them as like networks as well. So like this is the microbial population living in the copula on a certain date. So you can this would be like a genus, and you'd have all these different like species around it, for example. So we've got
this is the most gorgeous visualization. I guess if I can pull back a second from this amazing, what you're sharing with us is, what is this written in, and who's writing it, and is it open source? A little background on that.
This is just some stuff I knocked together. This particular one's from a D3 library.
D3.
Yeah. So it does that thing where you can like move it around, and it's all fun to play with.
Oh, I love it. And it really, the visualization really helps a lot of people like myself get it in context. Is it the back end code? You know if it's like Python or?
There's a combination of things. There's some HTML, Python. Wow. There's a few codes. What's the other one?
Mean to derail you.
Yeah. But so this visualization thing allows you to look at so previously, now this is another way of thinking abstract about data, right? Historically, what we're looking at those lists of microbes, And then we looked at lists with bars next to them showing us quantities. And then we were looking at like the different ways we can group organisms, like based on phylogenetic trees. And so there's these different ways of presenting the same data, but it just looks very differently. So this is that same kind of network that we just saw a second ago, or at least part of a bit bigger, but essentially bringing it all together in a kind of phylogenetic table. So this is like a dendrogram linking all the different microbes on the space station. So you can then like split it up based on, are you just interested in bacteria, archaea, going back through the evolutionary history. So this is a fun way of like exploring data visualization. So here, for example, we can see this branch where we get archaea separate from bacteria. So maybe we're interested in just looking at the data associated with this branch here, right? So I'm just going to drop dendrogram. That's archaea just there. So now we've applied a kind of filter thing, which allows us to see just the, what's it called? The archaea in these data sets. And as you can see here, this is the big underlying table. There's not a lot. You can see there's a lot of zeros, but 10, 10, 10. So the ISS HEPA filter, okay, is there. So I'm gonna come up here. There's only one archaea on the advanced resistance device at that moment. But if I go to the HEPA filter, what is it? The air filter, ISS, here we go. Let's check that one out. There we go. So we can see we get a few more. And there's a whole world of research people do about these ancient types of bacteria. And it's interesting to see that these really primitive, like single cell organisms that evolved before eukaryotes, before slime, before moss, before plants, for humans, these microbes are still here, these archaea, and they found a way to get up to the space station. It's awesome, isn't it?
Amazing. Okay,
so microbes are great. They're fascinating. No matter how old they are, they're always going to be there. No matter where we go, they're always going to be with us. They're not our enemy, they're our friends. Let's just be aware of them and do our best to make sure we have a healthy population, particularly if we're going to be doing things like eating lettuce and other organisms up there, because that will have its own microbiome too. And there's a fascinating paper that's just come out on that from Joya and co. But I'm very much a plant geneticist. So before leaving a lot of microbes, one last microbial game. So we've been talking about the selection pressures of the space station, right? So the space station is a stressful environment with radiation and weird gaseous mixing and all that stuff. Well, bacteria had been up there for 20 years, and so I was hinting that they might have started to evolve, or the community might have started to change. And what would happen if we put a microbe out there and look at its internal gene expression patterns? So we all have genes. Humans were about 20. How many genes we got? About 24,000. So Arabidopsis has about 25,000 genes on 5 chromosomes. So every organism, model organism, I've got a database that'll show me that one. It's quite embarrassing. I've got so every model organism has a total number of genes in its genomes, give or take a few. There's a bit of diversity. But what we're looking at here now is the bacterial model organism, Bacillus subtilis. And it's got about 12,000 genes on its bacterial genome. And what we're seeing here is 2 individual spaceflight experiments, one known as BRIC-21, one known as BRIC-23. I mentioned BRICS earlier, biological research in canister, so it's just a petri dish in a brick. And we see some really interesting patterns. Well, first of all here, so this is experiment 1, and this is experiment 2. Even though it's an identical experiment, when you look at the gene expression patterns, they cluster separately. So every time you send something to space, it's completely unique. Oh dear. Okay, great. Wow. And then we have here is those 2 samples. So this on the left here, we have our significance. So if something is more significant, it gets a smaller p-value. 0.05 is the general guaranteed cutoff point. Anything below 0.05 is deemed interesting and significant. So what I've gone and done is a negative log 10 transformation of that to make that really small number really big. So like the higher the number is up, the higher the dot, the more significant that sample is. So these are the most significant genes. I just selected the top 150.
Wonderful. That's such an amazing UI. This is like the dashboard of knowledge here you're sharing with us.
But the thing is, everybody that comes on here, if you give them the freedom to click on the side and select with the tools, what they'll all end up doing is selecting slightly different combinations, which is okay, that's fine. So what I've decided to do is just put in these filters here. So this just normalize it a little bit. Okay. So this is a 0.05 cutoff value. So we've just removed the dots that weren't significant from below, and we've used the genes that are significant in both studies. So if you look at the consensus of those two studies, you've got about 933 genes that were differentially expressed. So understanding the function of these genes is really important to understanding like how life is adapting to the spaceflight environment. And that's what these charts on the right here are designed to do. We have our genome ontology analysis and we have our pathway analysis. As we select genes on here, these update automatically. So we've got a few different Go types. So maybe I'm interested in like certain biological processes. So now we're selected down to a number of genes that have known biological processes. Maybe I think ion transport is going to be different and the cell wall organization is going to be different because of changes in the protein structure, right? Because of changes in the mechanical signaling because of the lack of gravity. So I now applied these filters based on logical deduction, right? And I think that's a really important feature there because now we've got down to 36 genes. When we were looking at 9,000 genes, I don't know what they do. No one's going to know what they all do. But like, if you can get down to a small number, you can start to give genes characters. Just like if you look at all the plants on the planet all at once, you've got so many, you can't imagine them all at once, right? But if you start to select subsets, you can start to give them patterns and start to give them characters and start to understand that one's a rose and another's a daffodil, and they have really distinct things associated with those characteristics. So the goal here now is saying genes have characteristics. We can give genes characteristics, and we can start to understand which groups of genes are responding to a specific environmental stress, like spaceflight. And these genes here would be the ones that would allow the microbes to evolve. These are the ways the microbes are adapting to that new environment that would allow it to pass on its genes to its progeny. Evolution in action. So there's no debate whether evolution is real. The Earth is not flat. So
can I ask how many generations before you start seeing that is an inherited trait, or is it immediate or unknown, or depending on the protein, or?
Yeah, good question.
There is no standard, right? Wow. Mm-hmm.
Yeah. So now we've gone pretty far. We've selected an interesting location and an interesting function. We found a whole bunch of like interesting biochemical pathways that have been altered. Okay, good. And you can see there's this button down here, this little green one. If you click on that, another button appears here. Yeah, this is the secret of the software. And if you click on that, you can then download the data. It will download the list of genes that you've just selected, their code names and their code symbols, their p-values, their fold change, all the things that the professors like to put into their research papers. It's just there. And it's easy as accessible as that. And at that point, there's one last little trick that you can do. And that is why on one of these sites, I put a link to string. Okay. So then what you do is you want to do genetic analysis, genetic network analysis. You see this pretty picture here. Okay, it's trying to load the database again. So there's a software called String, ShinyGo, NetMiner. These tools here, each of them you can click and plug out to, is essentially a place where you can just put a list of genes in, search, multiple proteins, you just stick a list of those genes in there. And then it will build a genetic network and give you all the statistics about it. The statistics that you want for your research paper, right?
I really have to ask, how has this scaled over time, right? Looking like a decade ago, these tools weren't available, were they?
Nope, not a decade ago, no.
This 5 years ago?
1 or 2 of them were available 5 years ago.
Is it kind of exponential then? I mean, our students right now in middle school or high school, if you're going to prognosticate, what kind of access to knowledge do you think they're going to have?
Well, I think this is the sort of thing they can have access to and do have the capacity to understand if they have a teacher explains it to them. For me, it wasn't available until I was a postdoc because the technology wasn't invented. But because of the advances in computer science, that not only has the technology been invented quickly, it's also been made accessible quickly. So when I was first accessing these tools, it was hidden behind walls of code. It was hard. And so I spent years trying to remove those walls of code and try and make it so you copy and paste a list of genes from one tool to the other, and then suddenly you've got statistical insight.
Perseverance, I think, is a key word there, right?
Yeah. And
what was the driver for you? What made you just say, I'm really going to have to get through this friction to get to the knowledge?
Well, just that there were so many patterns, and every time you find one and you go, oh, is that real? And once you get used to doing it, you start to believe those patterns. It takes a while before you get your confidence and start to believe the stuff you see. But then you realize, like, you can only convince other people if they discover it for themselves. There's this moment when you discover something and it's like, it's infectious. And so I realized by lowering the threshold to make it so these massive data sets are accessible by more people, they will look in there and they will see maybe some of the same patterns I saw and definitely some other ones.
Wow. You're like handing everyone a digital pan for gold, right? Teach them how to do that. Help them find those solutions.
Oh, I like that analogy. That's a nice one.
Extemporaneous, so you feel free to use it.
And every person's going to have a different sieve with a different size pores, and we'll pull out different patterns. And if they've got someone there to discuss it with, because this is why teachers need to be taught as well, right? Because they need someone to bounce the ideas off. This is really important in the scientific process. I said about how if we're going to push the boundaries of human knowledge, you don't do it in isolation by hitting your head against a cyber wall. You do it through conversation. You do it by pulling as many patterns together as you can and showing them to your friends and just saying, this is what I think. What do you think about it? Some of their perspectives may be different. Some of them might be contradictory, and some of them will be supportive and the same. And there's a balance between all of those things. And
Can I slip in a little advertisement there? The Leguminaut Challenge. Our next flight in August, you guys get to do that. We have a number of legumes. We need to determine which one we really want to fly. Dr. Barker here has some really deep data where we can kind of start digging in and discovering things like that. And for our younger students, we can just have some fun. But we'll be talking about that later in the afternoon. I just want to mention that. If it feels like it's a little deep water for some of your students, that's okay, because we're covering a whole gamut, anywhere from 5 to 99 plus, and whoever's interested in knowledge and access right now. So please, Dr. go on. It's really fascinating the way you take this and you make it accessible. These tools are really quite incredible. Is there any
Well, we've got a break there, Ted. We got a question from Facebook.
Oh, fantastic. What do you got? Go ahead.
Is it possible to incorporate this into a data analytics module with a future ExoLab or virtual classroom?
We will work on it.
Yeah, I guess we'd have to talk about APIs and things like that. With the deep knowledge that the repository that we're looking at right here, it would probably be recommended that we had high fidelity sets of data that met criteria for this, and that we then brought our data in and shared it with the collective, rather than us trying to do anything backwards from that. So wholly possible, I would say, but I would think we'd need to understand more about the filter that this data set requires. If you just put up, signal to noise ratio, if everyone just started throwing data in, at a certain point, it even becomes more difficult to find the patterns. And so what's the threshold, if you will? Am I kind of understanding that in the right way, Dr. Barker?
Indeed, and that's one of the things about your program is, like, if you get enough data, eventually you'll be able to fish out the good stuff, right? And your data is primarily imaging based, and so
we have imaging, and then we have some sensors. And you were
Right.
inspired when we chatted the other day to add even radiation, which we don't do. But those are relatively low power draw, and we can probably find a spot in our labs to do that, just to add another data point for you. But carbon dioxide, temp and humidity, and light, along with the imaging.
And so these genetic data, lots of these studies were done without any imaging, so we have no physiology data to go with it. So essentially, the goal would be for the students to go in, look at the genetic data, and then develop theories based on that, and see if they can be tested with physiological studies. So and So what I mean by physiology, this is, or can you see this video?
Right now we're looking at your data set.
My bad. Okay. 2 seconds. I'm just going to come out of this. I've switched over to another. Resume share. Stop. So I switched over to a different browser, and I'm trying to work out how to stop how to switch over to the different share.
Yeah, at the bottom of your screen, you might have something popping up, maybe a green box with an arrow.
Yeah, if I stop that share and then I share again, so this is the one I want. Okay, so physiological data is brilliant though, and you can learn so much from it. And so
Arabidopsis?
Yeah, so I'm in the process as part of the NASA GNAB program of trying to collect together all of the like physiology data of plants grown on the International Space Station. This is a data set that we've generated on Apex 5. So this is a plant on Earth, and this is a set of plants in space. Obviously, it's lagging, so I'll let that go. So we're trying to collect other images in the same kind of way. There's another one or two that's been done in the veggie growth system from Robert and Annalisa Poole. And obviously it's hanging up now, isn't it? Mm-hmm. So this is a plant growing on Earth, and it's great. We can see the roots go down towards gravity and away from like the light vector, and the plants look nice, happy, and healthy. Although when we remove the gravity vector, when we put plants on the space station, the roots just kind of go off all over the place. And when we start to measure these, we actually see they grow at a slower pace. So we're
should we go left? Should we go right? It's like they don't know.
You just go all over the place.
So light clearly, right? Phototropism is a driver as well. If you have light coming from just one direction, is the plant cheating on us?
Well, that's a really good question. So that's one of the things that from Rob and Annalisa's early work, there's a study done in the ABERS system where they had a row of LED lights just above the Petri dish, and the roots just happily grew away from that row of LED lights. Mm-hmm. And so that led a lot of people to believe that they didn't need the gravity vector to know to grow down, which was crazy. So the metadata of where you put the lights is really crucial, is really important. And also looking at like the direction that they grow. So this was a directional assessment that we did. So these are so I should go present. So we measured a load of different characteristics of their growth. Again, manuscript in prep, unpublished data, blah, blah, blah. But what we can see here is these are 4 different varieties, roots kind of growing down on Earth. And then here in the spaceflight environment, the roots kind of growing off in all different directions. You could also present that as a nightingale plot down the bottom here, the direction of growth. And the moral of the story was, it wasn't just like a perfect circle in the center. There was a downwards growth trajectory as a result of the lights. Oh crap, wrong button. Sorry, which one was that? Oh, I could just do this. That's way more sensible. Okay, cool. So meta-analysis data. Radiation data. Oh my god, where are we going? There we go. We spent a lot of time looking at these data, and so the directional information wasn't so informative. One time point wasn't so informative. The general pattern was flight routes were shorter. But when we took together the data from all the time points, it became a bit clearer. There was a subtle pattern. So like we saw them just growing off in all those random directions, and it's hard to conclude anything other than like, well, plants need gravity to give them a root vector. And that's going to be important to give them like a volume, which allows them to forage for nutrients and water. But what else could we learn with multiple time points? And we realized by measuring the speed of growth, we could learn something. So this is on the left axis. The y-axis, we have the total root system length. So you can see, given time on the bottom axis in days, over the multiple days, the roots got longer. Okay, things grew. So having students take photos of plants and measure things, be it leaf size or whatever, to practice making these graphs is an invaluable tool, and I urge you to do that. And it's really, really cool. In kinetic data is really important.
Right. I think next week we're going to have some folks from Fairchild and Trent Smith talking about growing beyond Earth and some of their projects with veggie, which are really fascinating, and being able to quantify, visual observation. Our little ExoLab has a scale on the side that students can.
So what happens when you live up there with cosmic rays? What happens? Was that a question? Yeah. What happens when humans live up there with cosmic rays? Well, the jury is still out. So generally speaking, your life expectancy is shortened. Yeah, how to put it? Yeah, the jury is out. Cosmic rays. Okay, here's a good study. So there's a group in Germany. Then they took the eye cells, stem cells for eyes for mice, and they grew them on a petri dish. So they have a nice flat surface and like a round petri dish. And then so there's cell cultures growing, and then they shoot a laser beam through it, a radiation beam. They used highly charged ions. I think it was like gold particles, but they had no electrons, and they just fired them from a particle accelerator in Brookhaven. And what it did is went straight through like a line. And you could see all of those cells because they're retina cells. When light goes through them, they kind of change in color and all this kind of thing. So it gave them the opportunity to do some interesting like imaging. And what they were able to do is basically show like, if you're in the direct beam, like that nucleus died, that cell died and had to like the cells next to divided to replace it. But the really beautiful thing was the fractals that came off it. As that initial sort of simulated galactic cosmic ray went through the petri dish, you saw these spirals come out of it. As all these like X-rays and protons were kind of fired out, and there's this gamma radiation creates these loops because you get X-rays, gamma, and like protons that kind of spin off it. It's the physics of it again is above my pay grade, but
and astronauts do say that they see that in their eyeball. They actually know when they've been hit by a cosmic ray. They're up there and they see this stuff. Chris Hadfield said exactly that.
Wow. Liam, do you know how often they have that kind of observation?
It didn't strike me as being something that was terribly frequent, but common enough that it was something that he managed to say happened multiple times.
Interesting.
Quite an extraordinary thing. Yeah.
Apparently it's more frequent near the moon as well. Like when the Apollo astronauts were out there, they're the first people to kind of noted it, and people then wondered, well, is it just because they're outside of the Van Allen belts, they're just that far out? But yeah, when Hadfield then said it too, people were like, oh no, the rays come in. In fact, NASA has a whole research program on the South Pole in collaboration with the National Science Foundation. Involves using high altitude balloons to essentially launch from McMurdo. They do a quick lap of the South Pole over about a week, and that's because that's where the magnetosphere comes in. So some of those intergalactic cosmic rays kind of get drawn into that magnetic well and can get much closer to Earth than they would usually. And so it could be a really interesting research opportunity to put like a life up into that area. And I wonder if we could see some flashes of light up there too.
just before you move away from the screen,
Go
for it.
Don't know whether Ted's frozen for everyone.
Oh, he's frozen.
Yeah. Anyway, what I was going to say, Richard, was I just got to note your mission patches are the best. Look at that one for toast, where you've got the actual.
In fact, I've got a good page for that.
There you go. Anyhow, and mission patches are an important part of every mission. And I've seen several of yours. You had a great one for the cotton sustainability project that you had, but that one that you had for the toast was quite phenomenal. Having the plants growing out of the toaster.
Yeah, it is a funny one.
And I know I'm sort of moving us away from the cool sciencey stuff here. But I couldn't help it.
No, but that's an important
this is
point, Liam. I mean, the arts are a way of expression, and if we didn't want human expression in all of this, we'd just send robotics missions. We wouldn't bother with human exploration.
You got it. But not everyone takes that up, and I just really wanted to acknowledge Gilroy Lab for deciding that was an important thing.
Hey, Liam, so I'll chime in here before we hand it back to the doctor. Is so for this mission coming up, Mission 8, teachers and parents, we do have a design challenge for the mission patch as well. So hang on later this afternoon and tomorrow and all next week as we get through 35 days of this collaboratory. We'll show you how you can do some design and bring some art into the experience as well.
Yeah. Art. Well, it's funny you should say that. It's so great. It's like as this, I have another version of this presentation which has all of the logos of the mission patches at the beginning, and obviously it won't load up right now because there
are too many mission patches.
But I have a little important thing about the whole STEAM component. I remember when I first went to a NASA conference and Darva Newman was presenting there. She was like the person in charge at the time, like the director of NASA. And she said we needed to put the A back in STEAM. And I thought that was really cool. She had a picture of the Starship Enterprise on the screen at the time. And I was really pleased because until that moment, there'd been a lot of downturn within NASA about the concept of teaching, and there'd been very much, this is a mission-driven organization, and the mission is Mars or the Moon, whatever the next president's whim is at the time. And so it was really nice to have someone at the top of the system turn around and say, no, we're about more than just the mission. We're about it, and part of the mission is inspiring the next generation. And there's and having that having someone acknowledging that arts are the way to do it. Like the engineers build the devices, the scientists come up with the experiments, but it's the artists, the visionaries that kind of painted the picture that inspired those other builders in the first place. I mean, it's not just about the economic and social impact, which this data visualization from the Canadian Space Agency really elegantly shows. Because if we just talk about that stuff, it's going to send everybody to sleep real quick. The whole point of space exploration is because it's exciting. It is because it's novel, and it's going to take humans on the next great voyage of exploration. Currently, we're right here. We're in the Earth Reliant phase. We're doing some amazing stuff with the help of the International Space Station and the Earth Life Science community. But we're going beyond there. We're going to go to the proving ground. We're going out to cislunar space. We're going to go to the moon. Project Artemis is going to take the next man and the first woman down to the lunar surface. And in the process of doing so, we'll be demonstrating some technologies that will take us beyond Earth Reliant. We'll be able to build the Deep Space Gateway, which will be a launching point for new experiments into deep space with the goal of sending humans to Mars. It's a really kind of exciting moment.
Wow, on Facebook, I just saw a bunch of thumbs up when you said go to Mars. It was like,
I mean, don't get me wrong, the International Space Station is awesome, and we shouldn't abandon it as a research community. It has laid the foundations, but the goal is beyond. It's beyond low Earth orbit, and is en route to Mars and a sustainable presence on the Moon. Mm-hmm. And so, with NASA building the sustainable steps based on von Braun's early strategy, it's great, but we do need private enterprise, and SpaceX and Blue Origins are going to bring some real game-changing technologies on this journey. And if we're going to do this, we need to do so sustainably. Project Artemis, as I mentioned earlier, is about not just going to the moon on a camping trip like we did in the Apollo era, which was awesome, but instead it's about going this sustainably and developing the technologies that allow us to go beyond the moon sustainably. And so now when we look to our ultimate goal, our ultimate destination of Mars, we start to think of Mars in a different light. I have a lot of students talk to me about planetary ecosystem synthesis or terraforming, as it's more commonly known. Mm-hmm. It's a really fascinating concept. Could humanity change a planet like Mars to make it habitable? And there's some great games in this area. Mars Terragenesis is a free one. You can download it. You can build all kinds of like terraforming projects to start to change like the planet as a whole.
And the name again? Can you say the name again?
Planet Terragenesis.
Terragenesis. So we'll find that link. We'll put it in there for folks to check out.
Yeah, it's like the first game like on Mars is free. It's one of those things that if you want to go on and terraform Venus, you have to pay a few bucks for. But I was able to complete the game on the free mode, so it's great.
So this makes me think, as a layperson, of what I call proto soil, because I don't know of a better word. If you're going to work with regolith on the surface of the moon, or with water ice, or on Mars, you've got that chemical composition. It's like your foundation. What are your thoughts on this idea of bootstrapping a soil, basically, this terraforming at a kind of a proof of concept scale? Could you get it in a little lab?
Yeah, I mean, it can be done. So FYI, The Martian, you've seen the movie?
Yep. Read the book, yeah.
And read the book, right? Okay, so again, steam's important. So can we grow plants in this Martian soil, in this regolith? So I learned something from Exolab. I was going through your material the other day. It turns out regolith is like just soil in the absence of biota.
Right.
So if we add biology to it, if we add organic matter, we can turn regolith.
Spark it, right?
Yeah.
Humans. Throw a lightning bolt on it, something. Right?
We are the gardeners, right, of the world. So what will this Martian soil? So you've seen the movie The Martian. He goes in and he uses his excrement, which he composts, and then uses it to grow his own food. Great. I'm not going to ruin the movie or book if you haven't read them or seen it. So, if you haven't seen them, you should go watch it this weekend. It's your homework, guys. But in this movie, he misses one thing. Okay, there's something impossible at the beginning. There's something impossible at the end. Okay, Hollywood, creative license, we'll give them that. But in the middle, it's not another one of these Hollywood movies where the US government spends too much money to save Matt Damon from some ridiculous situation he's got in. Matt Damon's not the star of this movie. The science is the star of this movie. That's why it's so good. And as he gets into all these little predicaments throughout his journey, it's the knowledge of science and how it works that gets him around and allows him to survive in this alien environment. There's one little bit that they miss, and it gets me, because, and it's probably just personally for me because it's a movie about a botanist, well, he is a botanist, but also an astrobotanist, and he misses one crucial detail, and that is he doesn't wash the soil. So all soil scientists know that Mars has got this thick layer of salt all over it. It's like not if the planet has like undergone desertification and salinization on a global scale. Lots of the water's been stripped away. So some of it's receded but a lot deeper. But then essentially you've left with this crisp crust of salt over the surface, which has then been bombarded by millions of years of UV, which has basically turned it into rocket fuel. So it is easy to remove. You've just got to wash it. So a few water rinses, he could have got rid of the salts, but he doesn't in the movie. And they do so many other things just right. And this thing.
That's amazing.
Yeah, according to some soil scientists that I had a conversation with, they calculated the water for what would actually the plants would need and everything, but they totally didn't take into consideration the amount of water that would need to be in the soil. So he's like several factors of whatever short. Now you add this additional one into it.
Yeah.
Well, there you go.
So close.
So we're still incredibly water expensive to even think about this. It's not just that we could plant our legumes in there and start building the nitrogen compounds, let those decompose and get some organic matter in there. We've got a tremendous water debt to deal with.
Yeah, that's always going to be the biggie. So this graphical representation, the
and not trying to put a downer on it for anybody. Just let them. These are the problems we got to have creative solutions to.
But you're right. It's going to come down to classic old-fashioned 4-course rotations, crop rotation systems. It's going to be good old-fashioned organic agriculture and things that hopefully people are practicing in their gardens this spring, like digging the ground, putting in like more organic matter. choosing the right plant species. You're right. It's going to be those fundamentals that need to be understood perfectly in order to get it to work on this alien habitat. But best of all, it's the same skill sets that we need to have in these urban environments. People often think that urban environments are not alien. They're the most alien habitat there is, like surrounding ourselves in concrete and metal and all that stuff. Like, so this is one of the other beautiful things about this new field of astrobotany is the way it's now feeding back into the urban agricultural movement. They're synonymous with one another. Like the massive urban factories of towers of plants growing up. Like that's how most of the graphic designers are painting the pictures of the lunar growth schemes and the Martian ones. Once we get past the we're here to camp stage, we get dug deep down into the ground. Why do I say that? We're mammals, right? Why are mammals the dominant? Why aren't dinosaurs, birds, whatever, dominant? Because when the great asteroid struck, we were digging down underground in burrows, like our mammalian precursors. Like, so humans. Well, this is an opinion at this point, right? This is why we're here. So when we go out into the stars, we're going to be digging down. Like, if you look at the radiation that's flying around, like the lunar surface, you do not want to be on the surface for long.
Those lava tubes. You can fit a city in some of those lava tubes.
Right? Why do you think Elon bought the Boring Company? Do you really think he wants to build a hyperloop underneath LA?
No. It is about, let me get a radiation shield going, some thermal insulation, right? Somewhere I can create a partial vacuum at least to get a little atmosphere going. Yeah. Why sit on top and get baked?
So again, so don't get me wrong. Like, I love the concept of growing the soil, like organic agriculture. Grow the soil, the plants will grow happily. But for the very first people that go there, all factors are currently pointing at hydroponics. And that's what this graphical rendering from the Kennedy Space Center shows with our friend Ray Wheeler. He's a good friend of Joya and Trent's. He is involved in the recent paper that came out examining the lettuce microbiome that was then consumed by the astronauts successfully and healthily. And so again, this is one of those great moments of art reflecting life, reflecting art. It's kind of like, so, yes. So you mentioned earlier the Fairchild Botanical Program. A huge fan of it. I just can't rave about it enough. And we're trying to do something similar with microgreens over the next few years. If anyone's interested, reach out. This was the other game I wanted to point out, SSI plants. So this is, again, totally free. It's from NASA. You can download it from your phone on an app. You fly around the space station in a very similar way to I demonstrated in the Mango app. And you have to get a few patches. You have to go to a few locations, so find a few things for the astronauts, like play along the game, and then eventually you can unlock the veggie growth system. And this is huge. As someone who spent the last 7 to 8 years working to eventually unlock the capacity to use the veggie growth system, it was really awesome to be able to do it in an afternoon on the game. And then what you can do is you can play with different intercropping systems. So there's tomatoes, lettuce, peppers, radishes. And you can do simulations as to what would happen if you have different water and wind levels, all that kind of the features that astronauts do change in the veggie growth system when growing vegetables. It's pretty good. So it's fun. I recommend playing that. And
the map you have there, I know it's expanded far beyond Florida, right? And I'm not sure
it's in Japan as well now.
Yeah. The next week, we're going to have the folks from Veggie join us and talk a little bit more about what this is all about. There's so many exciting ways to be involved with the International Space Station. We're, of course, sharing a wonderful afternoon here with Dr. Barker with microbes and plants, but there is, crystallography and microfluidics and material sciences and, genetics and epigenetics. And take the force of gravity out of the equation, what happens, right? So fascinating, and it gives us a different perception of what might be possible in these interactions, in this case of living things like microbes and plants. So we're going to have, over the next 35 days here, guests like Dr. Barker sharing these fascinating journeys with us. And actually, I hope, my goodness, that you come back for a session or two. I'm so inspired by what you shared, and if we can codify this in any way to inspire young folks that can't see this, we'll have to try to figure that out. What could you leave us with if you want to think about, as I don't want to eat in too much in your afternoon, you also have our entire afternoon if you want to join us with that, because I'm along for the ride, right? So I want to be cognizant of your time too.
How about we do a little bit of the rockets, and I'm no rocket scientist, so it's pure excitement. And then I'll finish off in the TOAST database, the genetics, to introduce like how students could use that to generate hypothesis to test in the ExoLab, something like that.
Sure, I love it.
Okay, cool. So as you can see, I'm a plant scientist. I'm constantly raving on about plants, right? But like if we're going to, but this whole spaceflight thing is kind of exciting. You get a lot of people who want to work with you because of it, which is fine. If they don't love plants, it's not their fault. They'll learn when they grow up. But some of them came to me and they're like, we just wanted the rocket science. And I didn't know about the rocket science. I was like, well, we just hitch a ride on SpaceX Falcons, right? Falcon 9. But like, so I had to play Kerbal to catch up. I love educational gaming, really. It's a fantastic way. You can learn a lot by playing. There's no reason learning isn't fun. And I learned that with Kerbal, it's really hard to do rocket science. I managed to crash into the moon, and I was so happy with that. And I was like, that'll do. So rocket science isn't
even getting close, right? Their expressions when it's
brilliant, right?
Microgravity. Oh, that's
So like so again, like if you're a teacher and play the game, you got some time off at the moment, and you will learn some things too, and it will help you be able to have the conversations with your students and get them interested in it. But fortunately, I don't have to do that now. The local high school rocket kids, they handle that area for me, because I'm more interested in in hitching rides on rockets. So we've used the Falcon 9 previously, and that's taken some stuff into orbit. And later on, supposedly next year, but who knows with the virus, maybe it'll probably be postponed. We're going to go up on the New Shepard, just get something suborbital to the edge of space for to look at that plant nervous system signaling, what happens with like 4 or 5 minutes microgravity, just like hanging out.
Yeah, I don't know if Erica Wagner from Blue is on, but if you are, Erica, your platform is fantastic. We can't wait to put something on Blue Origin mission.
Yeah, Erica's awesome.
I think are really nice. You only get about 4 minutes of microgravity, but you get those G's. It's kind of neat.
Interesting G transitions. So you have to be careful of like the experimental design, like where you're testing, but where there's a will, there's a way. And so this then got me appreciating the different classes of rockets. So we've got like the little hoppers. Falcon 9 is orbital. I've seen like the Blue Origins one, the suborbital one, in the factory. They had like 3 of them on the conveyor belt being built. So who knows where they're gonna go
on the conveyor belt?
Kinda, yeah. Like the different like cylinders, like bit by bit.
Yeah. There you go. Right. Amazon sticker on the side.
Delivery to the moon. Well, that's the whole point. Oh, that comes up shortly. And so that brings us to back to these new classes of rockets. Whoops. So here we've got the famous Saturn V. It's a great reference point, designed by von Braun to send humans to Mars, that was then used to send them to the moon. Is the SLS there? SLS, yeah. Well, this is probably the most expensive rocket ever built, and we'll see if
ever being built.
Ever being, right? It's a great workhorse. We'll see. We can't talk ill of it because it's the NASA program, and it'd be great to see the Orion fly on it. But the thing is, now in a new era with competition from private enterprise, and now with the big Falcon rocket, or now being rebranded as the Starship. So this next generation of SpaceX rocket is going to be huge. It's going to be comparable to Saturn V, and it's going to be designed to take people to Mars. But it'll be tested. It'll go through the proving ground on the moon. As will like the New Glenn, essentially. Although New Glenn has the lift required to get stuff to Mars, Jeff Bezos has said its purpose is just to build a delivery service to the moon. Some people say, "Oh, that's crazy. Who needs a delivery service to the moon?" Actually, there's already economic demand for it. Is it Toyota or Honda? That Japanese Toyota. Thank you.
You're talking about Japanese, the vehicle?
Toyota, yeah. They've got a lunar rover designed and built, and they want to send it up there, but there's no company they can just pay to take it. So the demand already exists for the economy in low Earth orbit. It's just and that's why Jeff Bezos has said he needs to build that, because the only reason Amazon exists is because the infrastructure he needed already existed within human civilization. So he says he now needs to build that infrastructure on the moon so the human civilization can leave the Earth, leave the cradle of humanity and transcend. But anyway, so rockets are awesome. They take our imaginations and human capacity to a whole new world. And we need to think about some of the people that painted the picture in the past. Have you guys heard of Zubrin before? He's a bit of a character too. So when George Bush Sr. was after a space program, he went to NASA and said, hey, tell me how to get to Mars. And so they had 90 days and they made a quick report. And they basically went to von Braun's old book and said, well, how would von Braun do it? And so they made a 90-day report. And it was, we're going to build a space station in low Earth orbit. We're going to build another space station near the moon. We're going to mine the moon. They're going to build a massive spaceship that's going to go to Mars. We'll already have another space station waiting at Mars. And then that will do reconnaissance missions down to the Martian surface for about 90 days. So you can go up. And they've said how much the whole thing was going to cost. And Congress looked at it, laughed, and threw it in the bin because it's an expensive endeavor. And people aren't often willing to plan 40 years ahead. But at this point, Boeing, Lockheed Martin, and like the Star Launch Alliance, they created like a monopoly. They formed the United Star Launch Alliance, and they provided the US government with military strategic launch capacity for like satellites and military stuff. And at the same time, they'd continue to develop like the von Braun strategy. We now have the space station. We now have self-landing rockets that was part of his plan. We now also are about to launch the Deep Space Gateway. The new space station is going to go near the moon. And recently, Boeing have just released their designs or the graphical renderings of the Martian space station that they envisaged building. So the initial plan to build the infrastructure to go to Mars was too much, too expensive, so they kind of pushed it aside. The aerospace companies kept developing the plans, could push it along slowly, piecemeal, one bit at a time. And this guy, Zubrin, said, you're all crazy, there's a better way to do it. Let's go Mars Direct. And that's what this book is. I recommend checking it out. And essentially he says, we can live off the land. We'll send a rocket there before. The rocket will use a biochemical thing known as a MOXIE that will extract the rocket fuel for the return journey from the atmosphere. And then eventually humans will go on the second or third rocket and we'll be able to have a rocket pre-prepared for their return journey that they will just go into, and essentially living off the land. You don't need all that extra infrastructure. You don't need the lunar base. All these different technologies that get developed along the way are superfluous if you go Mars Direct. In fact, there's some really funny videos of him shouting at Congress and getting really angry as he tries to explain to them they have to go now and they have to go direct.
I love passionate people.
Right? Me too. I mean, don't get me wrong. Like, I'm not saying we shouldn't shout at Congress. I think we need to shout them a lot more. Right. I'm just saying, like, when it comes from him in the 90s, in that way, he looked a bit crazy. But now we have Elon Musk saying, let's go Mars Direct. We have this billionaire, smooth as hell scientist, entrepreneur, who has invented self-landing rockets, who has made science fiction, science reality, and made a company called SpaceX, Space Exploration Technologies, with the sole mission of sending humans to Mars. Like, despite the millions he could make by floating it on the stock exchange, he's keeping sole control so he can ensure it achieves its mission. It's awesome. So the demonstration of the of the Tesla car was more than just like a brilliant advertising stunt for his electric car company, which is the we need to see in society right here, right now, to make sure we're sustainable here on Earth. It's awesome. So, but he's a sensible man, and this is what's so good about him. If you heard him talk, he's so cautious. He talks about probability streams. Oh, I love it. What a nerd. And he wants to go back to the moon. And here we have the first paying private customer, this Japanese billionaire who is going to go to the moon. You know, when I heard about his competition to take a bunch of artists with him Did you sign up? I've only, right? I couldn't think of anything that was good enough.
Well, everything you're doing right now is good enough. What are you talking about? You're at the highest level of creativity right now, Doctor.
Well, Well, to begin with, I thought you should do science. I'm like, oh, this is crazy. Why are they sending? They shouldn't send artists. They send scientists, take measurements and do all this stuff because science is the highest form of art. And I thought about it, let time pass, and we've thought about it again. I realized it's genius because the art that comes back is going to be priceless. Like, like science, we give it away. Like everything we learn and do, we have to give it away so other people can understand it and appreciate it. So it becomes the natural laws of science, right? Whereas the art, it becomes something which is intangibly valuable, something which is priceless, and things that like people flock to go see the Mona Lisa. And so when these
brilliant perspective,
right?
That's a brilliant
when these works of art come down from that first lunar flyby, it's going to create like a science boom across the world as these centers get placed, these art centers get placed. Beautiful.
Paint molecule has been, it has been placed in microgravity upon this campus.
Right. So nearly before I'm diving back to genetics, there's one last story which is obviously you can't not talk about Blue Origins, and their factories next to. So I was trying to put the scale of these rockets into perspective, and it's just impossible. But like this factory is next to Kennedy Space Center, and this thing down here, this is one of these massive trucks, and this thing is just huge. And every time I've gone back there in the last year, it's like 3 times. There's another building of this size that's gone down next to it. The infrastructure that Blue Origins, Amazon, Bezos, whatever is building here is huge. So this is just the beginning of the story. This is why it's such an exciting time. Within our lifetimes, we're going to see people go back to the moon, and they're going to land in ways that were only dreamt of in the Apollo era. We're going to be cautious to begin with because we're being rushed by the government, like Pence and Trump want to take credit for it. So they're saying, do it by 2024. Yeah. And everyone in NASA were like, oh, we were planning for 2028 because we're meant to be going sustainably with our European allies. And
we want to keep people alive during the mission.
Right? Right. There'd be a purpose. We've already got lunar rock.
Right.
The purpose is to begin and start building. Right. Because then ESA can land. Like ESA's got the plan to build there. So McMurdo is a fully functioning village right now on the South Pole. It takes, if we were to, okay, granted we can go on boats and stuff, but it takes longer to get to McMurdo than it does to get to the moon. So like, with the new generation of self-landing rockets, once Blue Origin comes into play, what are we going to do up there? The Lunar Olympics. Okay, we've got the Earth Olympics.
Hey, what will you do with 1/6 gravity, right?
Right. What sports will people invent? I don't know. Man. I can't wait to see them play them.
The long jump. Right. Basketball on the moon.
Suddenly, Quidditch becomes an Olympic sport.
Oh, there you go.
Right. So we're going to engineer the environment. We've got big lofty goals. At the moment, we're here. Okay, an LED rack with a shipping with a washing container around it, with a shower container around it, and little pillows with little plants. But we're going to get there. Like humanity have been nothing but tenacious, resilient. And so some people are trying to enhance the environment, trying to make it better for the plants and humans to live there. And I'm like, well, why don't we enhance the plants? Select the right varieties and make sure that they're being grown in the right way to enhance. And so this is a photo I took at the Kennedy Space Center a couple of years ago, looking at some NASA target crops. Look at that bok choy. I didn't even realize it comes in that purple variety. I should so get some of those
beautiful
things.
Color. Wow. Is that amaranth in the back, or is that all bok choy?
There's something different in the back. I'm not sure. But I just salivate every time I see that bok choy. Okay, cool. So more nutritious plants. Some friends of mine, actually, one last thing. So friends of mine in the John Innes Center, Genetics Research Institute in the UK, they genetically modified tomatoes. They put a gene from a petunia petal into the fruit of tomatoes, and it made them go black, deeper, darker purple. Those anthocyanins are antioxidants. They are really good for the anti-carcinogenic. They reduce your rates of cancer. So they proved this with the transgenic tomatoes that they gave them to mice that were genetically pre-programmed to get cancer and lowered their cancer rates. So eating colorful food is good for you. Eating locally grown food is good for you. It's more nutritious. So we want to select the right varieties to make sure the astronauts are healthy. But it's not just the quality; it's also quantity is an issue. So we've got to think of harvest index. So what proportion of the plant do you eat? So when we were looking at the bok choy earlier and the lettuce earlier, they've got a really high harvest index. So you eat most of the plant. You don't really eat roots, but you eat the rest. So on the left here, this is the world's cereal production and the world's population. You can see as human growth, population growth has gone exponential, our capacity to produce food on the agro ecosystems that we had has also gone exponential. And that's because of what we call the green revolution. That's the industrialization, mechanization and, of agriculture. And what that was made possible by these mutant varieties. You see these different plants here getting smaller. This is the reduced height allele. In wheat, it's a mutation in a gene called a DELA protein. Don't get me started on that. That was my dissertation. But the more of the story is the reason civilization is as big as it is now is because of these dwarf varieties and the domestication of plants by humanity. So if humanity is to have a sustainable presence out amongst the stars, on the moon and beyond, we're going to need to take some of these modern strains, these dwarf varieties with us. And so we need to understand how they adapt to the spaceflight environment.
It's kind of funny. Our first astronauts were small, right?
Mm-hmm.
As a requirement for the capsules. Just a point. Yeah. No connection whatsoever.
So actually, my old supervisor, my PhD boss supervisor, he's actually created some new dwarf varieties. So if you want, NASA has flown some dwarf wheat varieties already. But if you ever want to go in that direction, let me know.
Yes. My answer is usually yes.
But then this takes us back to, so wheat is a really applied thing. But most of my studies I do in Arabidopsis. And this is not going to work. Okay. And so why Arabidopsis? So Arabidopsis thaliana is a small, fast-growing weed. It's a member of the Brassicaceae family. So it's a close relative of all the vegetables. Broccoli and stuff like that, cauliflower. And so it's been a focus of research for a lot of labs around the world. We know everything about its genome. It's got 25,000 genes, and we know the function of a lot of those genes. And so there's been a collaborative international effort to understand this plant's cellular, molecular, and genetic data. And then we can look at natural variation of how it responds. So these lines represent 4 different varieties of Arabidopsis. And we've done a gravitropism assay. So we've turned the plants on their side and the root bends down and the shoot will bend up. And then we can take time-lapse photography and measure the angles at the tips over time and see them respond to the new gravity vector. And basically, depending on some varieties are more sensitive to gravity and some are less sensitive.
And just to be clear, if I can, this can be done in the classroom.
Oh yeah.
Right? It's simple as a rotation of 90 degrees. Okay.
And we did this with, give me a second. We did this with 60, I did this with 60 different varieties of Brachypodium, a grass, close relative of rice and wheat. And we used that data to persuade NASA to allow us to send 3 varieties of Brachypodium to the space station. So understanding natural variation is a really interesting way to start research programs with NASA. And this particular one here, this is with Arabidopsis. So my first spaceflight study my second was one with Arabidopsis. My second spaceflight study with Arabidopsis was with natural variation, looking at these 4 different varieties. There's an American, a Polish, a Spanish, and a German variety there. I'm sure there's a joke where they all walk into a bar. I just haven't worked it out. But the more of the story is you can see natural variation and sensitivity to gravity. And so if we're trying to engineer like plants for the space environment, perhaps we should just understand the natural variation that already occurs and then select some studies based on that. So let's show you. We could do the launch. I was gonna say, because we could dive into the genetics, but I should just stick to the story.
Take us wherever you want to go. I'm just really enraptured. This is fantastic. Thank you so much, Dr. Hook.
So okay, so we talked about the natural variation sensitivity to gravity, and we talked about the plant nervous system earlier. I showed you that video. So what about variation to how they sense the spaceflight environment? There's natural variation in that, right? You can do it with pretty much any stress. Okay, let's have a take another look at different things. Go back to, oh, not that other way. It's not the one. There's not the one I am looking for. Here we go. Sources. It's the, you know, this bloody thing pops up in the screen above you and it conceals the button you want. It's right there.
You're looking at all the images of the folks in the chat. You can grab that box and pull it down to the bottom too if you want. Or off
makes
to the
sense.
side.
So this is a cool. So this is a natural variation study. So we showed you earlier the plant nervous system responding to glutamate and to wounding. This is the same thing responding to salt. So I played the video and I'll play it again because it's pretty fast. So we're seeing a yellow, and now look on the left for red. Look for a reddy orange. There. Still there? So that's just a way of imaging that same calcium wave. We're just using a different sensor in this time. So we're adding salt at the end of the route, and then an electrical wave is going on the plant, telling us the plant's encountered salt. So then what we start to do is look at the natural variation in plants' response to salt. So each of these X's represents a different place on the globe where Arabidopsis has been found. And I was able to get the seeds for about 100 varieties, 108. And so now we have the seeds and the GPS coordinates of where those were. They have different names, these silly code names that tell you where they're from. Okay, great. And this is their growth on salt. So some of them are bigger and then some of them are smaller. So if they're smaller, they're more sensitive to the salinity stress. And if they're bigger, they're actually less sensitive to it or kind of like it. So now what we can do is we can now think of this as, instead of salt, think of this as, say, gravity or something associated with gravity like radiation or low oxygen. Now what we've been able to do is we've assessed the global population and found the varieties that are most resistant to that stress.
Wow, brackish water around the world and food.
So you can see the global terrestrial impact of this particular research tangent here. I can see, but you can also see how you can switch out something as terrifying as salinity in agriculture with gravity or response to gravity, and then you have a spaceflight study. And anyhow, so I'm not gonna play this bit anymore. You can zoom in and see where they are and all that jazz. But okay, so natural variation. So I was introducing this because we did a natural variation study on the space station. We weren't able to grow 100 or so different varieties. We just don't have the manpower for that. If only we knew a place where there were hundreds of students who wanted to do experiments to collect big data sets.
I wonder.
And then, so we flew 4 varieties and we put them on the space station and we asked what's their natural response. So microgravity. So we're just going to look at one variety to begin with, I think. So now we've got plants in microgravity. Got to have the video of Chris Hadfield playing with water because it's awesome. So, because again, we're trying to put the genetic data that's now appearing below here into context. So what do you think is going to be the most stressful thing about space? Like, what are the sort of patterns we're going to be searching for in the genetics? Well, radiation, but like galactic cosmic rays, nothing's really encountered that before, so we don't really know what that pattern is going to look like. Now imagine trying to water your plant. So now imagine this rag here is your plant. Your plant has to breathe. It needs oxygen. It's a living thing like you and I. And now when you're watering it, you're coating it in water. And even when you don't have water there, it breathing is creating that bubble of CO2 around it, right? So can we find patterns of CO2? Can we find patterns of low oxygen? Maybe. Okay, so scrolling down. This is one of those data visualizations that we made. Whoops. Okay, cool. So push this up a little bit. Push this to the side. Okay. So here we have the genetic response. So this was GODS 117. So, what's that? I can click on here and it's going to pop open into a new screen. So this is your background metadata. And so throughout these apps, I've put links to NASA GeneLab, where the place I pulled the data from. And here you can go in and you can look and read how they did the study. It has all this associated metadata. They don't have sensors quite as good as yours, but they've done a pretty good job at collecting as much as they can. And here's the research paper associated with it, etc. So these genes are responding to the spaceflight environment. So this is our significance along the side. This is the standard deviation. So I'm just going to select the genes that are most significant. And now we
can also select for deviation as well across the other axis. Oh, okay.
Exactly that, yeah. So this was just one little, and then we've selected those 61 genes. I can go up here, and these things here now telling me their function. At the moment, this is all in this mini box. This button here will make it pop out, make it go into a full screen. Okay, so the more so that's a tiny snapshot of toast, and I'm just going to show you what toast is again. Again, this video here is got just kind of it's like just help you fly through toast. It's got my crazy electro music over the top, but I'd rather just kind of play with it and show you. So this thing here are those different dashboards. Each of these is a different spaceflight study. If you've never sent an experiment to a space station, I recommend watching this little animation. It's made by NASA Ames, and it shows you all this pre-steps that you go through in preparation. It's actually really good. Because I watched it before going through the procedures, and then I went through them, and I was familiar as I went through them because of this animation. So it is legit. And so each of these, again, different studies kind of picked from, and we'll show you how to do that in a moment or two. At the bottom, these are other bioinformatic tools, other computational tools that bring data associated with Arabidopsis to you. So once you've mined the data about Arabidopsis in outer space, you can then compare and contrast and link that to other Arabidopsis research resources. These things, again, weren't around a few years ago. And I've just been collecting the best and embedding them and putting them together to be like a one-stop resource for someone who's never heard of Arabidopsis, but wants to get the latest, easiest to use tools. And then, as mentioned previously, if you can pull out gene lists from these things, and down the bottom here, I've hidden links to some extra like statistical tools for those who will love their biostatistics. Who doesn't, right? Okay, cool. So I'm just going to launch it into my next browser, full screen. So opens up, we're in. I'm going to go to my favorite study, the one that I previously mentioned, one of these two ones here. I mentioned the natural variation. So I've just changed it from a grid view to a menu. We have a bunch of extra metadata describing some of the key features of how the experiment was conducted. Just scrolling down. Ooh, here's my coal wild type. That's my favorite. These ones here. GODS 37, GODS 37, A, B. There's so much data, I had to split it into two different dashboards. So this is what we call colloquially BRIC 19. As an experiment, we grew 4 different varieties of Arabidopsis. You guys have flown Columbia, if I'm not mistaken, previously?
That's correct.
And WS is the other major kind of model organism. They're the two that have flown the most. Yeah, different ecotypes. Columbia, Missouri. WS is someplace from Poland that I can't pronounce. Okay, cool. So we've got about 33,000 total like ensemble transcript IDs, gene IDs. Okay, cool. Lots of genes. These buttons here change through. We can look at the biological processes, the molecular functions, and the subcellular localization that these genes here are involved in. At the moment, we've got all of them selected. So this is like the total, all known knowledge about all of the subcellular localizations and molecular function, etc.
It's kind of an interesting distribution curve there. You've got those kind of bimodal bubbles at each edge, right? And then you've got obviously the column in the center. But there seems to be some gaps, or is that just maybe I just don't have high enough resolution?
I'm not sure. Was it looking at these guys?
Yeah, just looking at the top left graph there, called flight to ground versus ground.
Oh, these things here.
Yeah, so you've got the large center column of points, and then it looks like some kind of a bimodal edge on each side of that column.
Exactly. Yeah, I'm with you now. Yep. So so this is like what we call the process data. This is what geneticists like my professor Simon salivate over. So this is like this final spread of the data. So this is all 34,000 genes responding all at once in one graph. As we go to the right here, these are genes that are going up in abundance in spaceflight. As we go to the left, these are genes that are being switched off in response to spaceflight.
And the ones in center essentially are saying, hey, what's going on? Nothing. I don't feel anything.
Yeah, pretty much subtle changes.
Wow.
And as we go up, we're getting more significant. So we've done a negative log 10 transformation of the p-value. So the higher it is, the more significant it is.
So am I understanding correctly the no change in the center at the top? Or
so you can have like really subtle change and it still be significant if the gene is really highly expressed.
Mm-hmm.
So I'm just gonna. So
these are all the changes that you see more of, right? Interesting.
So these are all those genes. Close that down a bit. Okay. And so on the side here now, we have this table, and this has those genes I've just selected. We're filtered down, and this has the locus IDs, the transcript IDs, the gene name, and like the expression level during this study all next to it. So I did that really manually, but I can also do it just by using this pre-made filter. So going off to 0.05. So this is
no. Like the binary.
Like significant, yes. That's my kind of significance test. Right?
Do you have a quantum button? Can you press that?
If only, right? So what we've done here, we've got these two varieties, the Polish and the American, and we've selected genes that are significant in both of them. So this is like the overlap. Maybe we're just interested in the genes that went up in one of them, and maybe the genes in both of them. So now we've sub-selected the genes based on significance between two varieties of Arabidopsis and genes that went up. And now we can go out and we can start to look at like their subcellular localization. I was showing to someone the other day, and they were saying that they think the plasma membrane encounters like some weird mechanical forces because there's no gravity. And now we've gone and selected those 6 genes that are in the plasma membrane that responded in both those distinct varieties from different places in the world, and both went up. So maybe these are the genes that are being activated by the plants in order to help them adapt their membranes to the lack of gravity. So we've generated a hypothesis here, and now it needs to be tested. It needs more work to be done. So there's a green button in the bottom. You click on that. You can then download that list of genes with that data. And that list of genes with that data, you can then put into you can then put into those tools that I mentioned earlier. The string tool, put them in there. Or those other ones that right down the bottom here, ShinyGo and Netminer, which will give you links to all of the literature and give you statistics to tell you which pieces of literature are enriched. This is how you find out the function of these groups of genes. This is modern data mining. But this is just looking at one study. Why don't I look at another study? Because this is linked to all the other studies that have genetic data in the public domain at the moment. As far as I'm aware. Okay, maybe I've missed one, but we'll see.
Absolutely fabulous.
So this was done with a study using RNA-seq. So I'm going to this next study is done with us using a microarray. Can you see those dots? So RNA-seq monitors every genes. Microarray only monitors about 24,000. It was an older technology. So I don't know if these 6 genes are on those old microarrays. That's my problem. So look, you can see it's not there. So so that's unfortunate, but that's just the nature of the technology. And this this is using the Brookhaven National Laboratory. So, and I was filtering based on plasma membrane because I was looking for something in response to gravity, but now I'm looking at a radiation data set. So what do we think would happen in response to radiation? So we would probably get something associated with DNA binding, maybe? Transporter.
If there are any students that are watching, homeschoolers or parents, your children have thought the internet is always around just for movies, right? or for social networking, right? CERN and the World Wide Web was all about, can we get together and share some knowledge, right? And here's these amazing tools What a journey you're sending us on, Dr. Barker. Thank you so much.
Well, hopefully you can see that the way I see this, it's just a computer game that I've made. And if other people come play the computer game, they will find other patterns within it. And then we can test all those patterns together. Like there's so many patterns in here. So like, here you go, prox days. So based on the molecular function, when a radiation particle goes through a living thing, it splits oxygen and it creates reactive oxygen species. And that's why people say that it's good to eat food which is colorful because it's high in antioxidants, right? So I basically just use a very simple piece of science that everyone knows to select genes of known function that would be involved in the antioxidant response. And here we are now looking at a Brookhaven study. So 1.5 hours after being shot by a particle accelerator, these genes were activated. Those same genes were also activated or changed by this, activated by the spaceflight environment in those two different varieties. So this is how we generate hypotheses. We now have a theory saying that maybe these genes are involved in radiation response, based and not just on Earth in a particle accelerator, as done here by the Brookhaven study. Again, like the paper associated with it is just hidden to the side there. But also in the context of the spaceflight, because we put those filters on based on that previous spaceflight study. Right. So, and I've been doing this over and over, and I get really excited. Can you tell? I get really excited. But like ultimately, we now need other people to basically play the same game and then record this. So one then downloads the list of genes, one then puts those genes through a couple of other tools, have a quick play with these. And then what I do is I put them in those Google Slides. And I showed you some Google slideshows earlier. And then how essentially we end up just like fishing through these genetic networks, trying to find like patterns that make sense in the data. Like I think a lot of times the media gives science like a bad rep for being something which is like boring. But like once you get into like the depths of it and you start fishing through, and then suddenly patterns pop out, it's a really fulfilling pastime. And that is why I feel really lucky to have this opportunity to share this passion and share these data with you guys today. With the hope that maybe you want to come back, revisit them, and help mine these data to help make some stories of your own. So, so finally, so the final tangent of the story. Okay, cool. So I gave you a quick introduction. This is Toast. The data is here. It's everybody's data. If you need a hand getting to understand using it or put more points on it, let me know. You can see it's just a menu. There's a lot to choose from. It really is a question generation, hypothesis generation machine. It's also a great place because I've centralized all the literature and made sure it's not behind paywalls, so anybody can access it.
Dr. Barker, what we'd like to do post this collaboratory here is take and provide a primer on this and put it on our platform. That way any educator can come in and use this as a resource. And if we can actually get them on ramp fairly easily and get right into the meat of what you've offered here, it's fantastic.
Mm-hmm. And I think that'd be excellent. And so hopefully, I think I said this a couple of times, but my real goal here, as you can see, like, 1 second. So let me show you something, 2 seconds. Okay, so this one, these ones, not that one, this one, they were all done on the space station with no photography. We have the genetics, but no visual imaging of what the plants looked like when they went through that. If anybody goes into these studies and they pull out a pattern that they think they can link back to the genetics that controls the size and the shapes of the plants, then that creates a new opportunity for other spaceflight studies where plant imaging capacity is possible. You could imagine trying to replicate these studies as true to their original form, and then flying them in a system that has a camera to provide that imaging data that wasn't available in these early days of spaceflight studies. That's just a subtle hint, eh? You see the angles, right?
I've taken it. Yeah, absolutely. We'd be honored to find a way to collaborate, even on the edge, very edge.
Like that one there, that didn't have any photography. Nor did that. Anyway, okay, I digress. So, hypothesis generation machine right there, entirely focused on plants. And then because we did it with plants, we went one further. So there's always going to be someone in your class who doesn't like plants. It's not right. It's not their fault. They're only human. We're not as advanced as plants. So we do make two stupid things. So thanks to the support from NASA Gene Lab, we've had the opportunity to do it again. So this, there's a certain filter that's been applied. So we don't have quite as many plant data sets here. I had to remove some of them because they didn't conform to NASA's requirements, basically. But so I removed a few plants, but I added loads of human data and rodent data and Drosophila and the yeast and such. And I've linked it in the back end. This is the really cool thing. I've linked it in the back end, so you can select genes in, say, a mouse, and then see what the related genes in humans are doing. What? Wow. So we're all kind of related through an evolutionary tree, right? Before showing you that, I'll just quickly do the backstory. Orthology. So we're all linked through an evolutionary tree. As we go back in time, we can see that. There's many ways to visualize the tree of life. 5, 4, 3, 2, 1. Some of them can look really pretty, like this, any moment now. You get a lovely big circle that shows you the tree of life. Okay, so it'll come in a second. There it is. So that's the tree of life as we currently know it. So each of these ends here is a different kind of species. And we can link through, by looking at the genomes, we can see how we all come basically back to an early sort of ancestral cells, right? It's kind of cool. And this thing here, you can play with it. There's a lot of information here on how to make that look different and better and export it. Thanks to our friends at the Tree of Life project. But hang on. So there's many ways to think of the tree of life. So this is another couple of graphical representations. So we have fish, mammals, all these things going back to our like early ancestors before the Cambrian explosion. Similarly here, another way of visualizing that tree of life. I kind of like this one. Do you recognize it? Yeah, so this is how Darwin first sketched the first tree of life when he first thought of the theory of evolution back in the day. So as we go out into the stars, what evolution may or may not occur? Can we adapt? So that's why, so we've gone in, we pulled in the data from these different model organisms, and we've linked them based on their orthology, their related genes, and provided the same kind of, okay, cool. And provided the same kind of, I'll start back again. And provide the same kind of interactive system that allows you to compare the studies between each other. And so it's slightly different to the previous one, but basically the same. So I'll show you it in action. Who would do anything other than plants anyway? Crazy people. So like, I'm gonna say, I'm not gonna use the rodent data today. There's a lag because currently it loads up. There's so much data in the rodent area. So the Gene Lab program, they had the remit to make loads of spaceflight data. So they flew a bunch of rodents, and in fact, yeah, let's load this up into full screen. So they flew a load of rodents and they cut them into little pieces. And so they got data from the livers, from the muscles, from the fibula, all these different places. So let's have a quick look at the Gene Lab as a whole. So NASA Gene Lab, I mentioned it earlier, it's a big repository, and it's got over 216 data sets. It's got over 146 different factors, over 33 different types of species. And when you look at species analysis methods, a lot. So there's a lot of variation in the way studies have been done in spaceflight. And you can see mouse is number 1, and humans, then Arabidopsis is number 3. It's good, I'll take that. Okay, cool. The way most people monitor plant or life response to spaceflight is transcriptional profiling. That genetics that I was showing you earlier, this is transcriptional profiling. And there's many different factors that are investigated. Spaceflight's difficult, you can't isolate one factor. So even this thing here where they say microgravity is a gross oversimplification. And so you have to go into the papers and search through and try and add more metadata factors. And then we can create these big tables, and we can start to search for GeneLab accessions. So these codes here, these are code names for studies that have been done on like human stem cells, when they looked at their genetics, their transcriptional profiling, and using simulated microgravity. Here's a link to the paper and the data, blah, blah, blah. Okay, cool. A bit boring, I know, I'm sorry. But I spend my life going through these tables of data and trying to make it look pretty. So these are all those different studies. We know a bunch of the different factors. We can start to group them based on the technologies, the species, and the analysis methods. And so we're trying to oversimplify all this. There's so much data here. How do we focus in and find one study that we can look at? So obviously we'd filter, we're going to use a Arabidopsis. Well, that made our life easier. Way. Okay, cool. And then we can do it based on like maybe transcriptional profiling. Because again, the main technology, like stick to the mainstream. Trust me, it's a good idea. So here we have all the list of all the studies, the factors, and so now we have all these different factors. Ooh, that's what I like, factors. Okay, cool. Cosmic radiation, organ parts. Okay, cool. 2, 2, 3, etc. Okay, great. So based on like the metadata, I'm starting to see a few studies that I'm interested in. I'm seeing the cosmic radiation 121 or like that. Okay, cool. So I'm interested in something with cosmic radiation. Okay, that's cool. So now I'm going to go back. I've got this drop down chart again here. Ooh, now I'm back in my menu. Okay, so I mentioned earlier, well, there's all these different organisms to choose from, but I'm going to go to plants because I like plants, and I'm too impatient to wait for mice. Okay, and I'm going to get rid of these two filters because I know the sort of study I'm looking for. This is a transcriptional profiling study. Okay.
So I just want to mention here, a little editorializing. So, folks, this is how scientists really work. That thing that the scientific model, and it starts here and goes there, and it's a non-linear process. And Richard, I think you're doing a fabulous job of showing how you can go down some the craziest rabbit holes and find some really interesting things. I think we all got to be Alice and just go down the rabbit hole and see what happens.
Precisely that. And the more people go down more rabbit holes, the better. Like, because the greatest supercomputers in the world are in the heads of the students in the classrooms. Problem is, they've only got TikTok to entertain them. Sorry. Maybe, but
sorry, not sorry.
Yeah. So this is why we wanted to put these data out here, because again, the coding that went before this can put a lot of people off. And there's a lot to be said for like learning Python and R and these things. But everyone wants to do the biostatistics. Some people like me were biologists. So this is a system that's been made biologist friendly. And I think that's quite a large area, proportion.
Where were you 30 years ago? Because I spent hours and hours and hours going through the ERIC journals, just looking for a reference.
Mm-hmm. It's hard, but now for these guys, you click here and the reference is there. Okay, so anyway. So yeah, this is just that link out to the NASA Gene Lab where the papers and all the metadata is. So this is the first kind of summary. So here we've got in a row, we've got 1, 2, 3, 4, 5, 6, 7, like 10 different plant studies in a row, right? And one of them earlier, I saw it said something about ionizing radiation. That was 121. So I used that metadata, 120, that's the one. I used that metadata app earlier to identify these studies as being potentially of interest. This one, I've done something funny, I clicked on the screen, now it's zoomed in a bit too much. So because I clicked on that, I'm not gonna use that one, I'm gonna use this one. So you can put the filter, so you can see using the significance again. I'm gonna just select those genes there. So it's gonna take a moment to load because it's like now gonna update all of the graphs here. So now I can go up and down. It's got a couple on that one. Maybe I think select.
I'm looking at the title of the graph. I mean, that root tips in light and dark. That seems like a very accessible experiment.
Yeah. That was okay, we'll go back to that one. Okay, so root tips, cell wall organization, root tips in light and dark. So it's this one here. Okay, cool. So so we can use this thing here to filter on this one graph, but this is an oversimplification. This graph is summarized plants that are grown in light and in dark. And that's what these buttons are here for. So I'm just going to click on that. So we're now gone to a different place on the database, and we're now looking at that study in a bit more detail. So we still have the filter that we applied on the previous study. On a bigger screen, this thing here, actually, I do Command minus. There you go. So sometimes these buttons aren't readable because these are like drop-down menus. So you can again apply all the filters here. Still got the 9 genes we selected previously. We can see what they're doing here, but I'm gonna turn off that filter. Oh no, look, there's a typo. I should put a space in there. God damn it. Okay, cool. So this was this is one study. Actually, this isn't the light and dark, this is the next one up. But you can see here, this is that volcano plot we introduced earlier. Nice overview. This is the GeneLab summary of the p-value and the standard deviation of the loci. This is a pairwise comparison of ground versus flight. So we can see genes going up, genes going down. And so this is a really kind of like simple experimental design. You can see there's different ways of looking at these data. We can again select the most significant ones in a really kind of crude way and start to use this table here to start to give them descriptions. So, auxin. Auxin is moved through the plants in a polar fashion, directed by gravity. Expansins. That's involved in cell wall loosening. Another expansin. There's a paper that shows cell wall structures altered in spaceflight. So these are probably the genes that regulate that. Workies are stress response proteins. We see them coming up in a lot of spaceflight studies, probably as a result of low oxygen. Scarecrow-like 13, that's actually one of my favorite genes of all time. I know, I'm a nerd. But
Scarecrow 13. I guess the discoverer of it gets to call it what they want, kind of like asteroids or something.
Exactly that, yeah. So Scarecrow, it functions in a pathway with magpie and jackdaw. So geneticists have a sense of humor too. It may not be very good, but okay, cool. So that's the sort of thing again that the old-fashioned geneticists love. Download, put into the statistical tool. That's the story. So I'm just going to go to the light and dark study earlier that you mentioned a second ago, because we selected 42 genes from this BRIC16 study that had altered cell wall signaling. Coming down to the plant section, etiolated seedling. There it is. 121 versus 120. My bad. So 5, 4, 3, 2, 1. Okay. So they were the top 41 genes that were selected in the previous study, where they have a paper that they write about alterations to the cell walls. And now here, looking at light and dark. Oh, wow. Live demos are a sure way to find potentially mistaken the code. I wonder. That should be colorful, but who cares? You can see those 2 genes there and there. I mean, they were really jumping out. So at this moment, I want to know what they do. Okay. Okay, cool. FOT1. That's one of the light receptors, blue light receptor. Isn't that awesome? Okay, cool. So
that's quite amazing, actually.
I'm not sure if you can see. I'm genuinely excited about it because it's like you would hypothesize, you would postulate that if you've got plants in light and dark, and if you're removing gravity, the capacity to perceive light would be different between them, right? And here we've got one of these the core light response light receptors, FOT1, being differentially expressed. Anyway, I digress.
More sensitive in space, is that correct? You said this is the stuff we pulled from the right on the earlier graph?
Yeah. So again,
does that mean less sensitive, or?
I would have to look at the fold change, and then you'd induce it from you'd assume it from that.
Very fascinating, but it is a significant change. Is the most important thing I think you want to point out here.
Exactly that. 780.
I've been creating a list of questions for you too, by the way, Doctor. So
Really?
I just don't want to
rabbit hole to actually chase.
Yeah, there you go. Right? Do you feel like the Greyhound there, Mr. Wilkinson?
Well, that, or I'm just thinking of the Turing machines. Figuring out which equations are worth, or problems are even worth pursuing. Because is there even anything of interest there?
I'm shuffling to the discount bin right now. I have no idea what I'm looking for.
Right. So the final thing. So now, because of this, I've got this FOT1, and there's a tool that is embedded in ePlant. They're called ePlant. I'm not sure if I still have it in here, if I took out the new one. So in the original Toast, it's down here, but in this GNAB Cross Species Explorer, it's not here anymore because it's for all species. But I'm just going to type in the term ePlant. It stands for electronic plant. I think I showed you some people this previously. But finally, like, I found this gene, FOT1. I want to know what it does. And so I can literally just type the name in here. And it's now going to go through databases of hundreds of studies, and it's going to produce pretty pictures that tell me what this gene does. A picture says 1,000 words, right?
It sure does. And for all of those that kind of popped in during midstream here, Dr. Barker's taken us on a wild ride from the microbes to the plants to space to rockets to life beyond Earth to life here on Earth. Absolutely fascinating. And we're going to try to create some annotations with some links of all the this massive amount of this wealth of information that will be there at your fingertips. All ages of students, and lifelong learners.
Yes, quite, because I'm a lifelong learner. You never stop learning, and I've been collecting all this stuff together to try and help other people like mine these data and to like collaborate with each other. So and that's kind of why I'm really excited to have this opportunity to share these methods and tools. So you can see here, like, it speaks volumes. There it is. So the light receptor is in the plasma membrane. It's where plants perceive light, blue light. And so we've stumbled on something that a really interesting concept. Without gravity, your blue light receptor is altered. And when you grow plants in light and dark, the blue light receptor is altered. And that's kind of why this particular gene kind of popped out. So the difference between a plant being grown in light and dark and changing its capacity to perceive light, that is obvious, I believe. I think that's a fair thing that hypothesis that you'd have just based on, if I'm putting this plant in darkness, it's going to change its light perception system, right? Saying I'm going to put the plant in microgravity, why would that make you change your blue light reception system? I mean, this is in the data. This is the little pattern that we've just discovered with this simple comparison.
Why would that?
Mm-hmm. Well, on Earth, whenever you've seed is germ, again, I don't have an honest answer, but I've got a little bit of a story that help put it in perspective. So on Earth, when a seed germinates, roots grow down towards water, shoots go up towards light. If you do it in a dark box, shoot still goes up, root still goes down, because in the organism, there's a genetic mechanism that allows them to perceive up from down. So in the absence, but when you have light is the next major feature. So the thing that controls the plant's orientation is always a sort of environmental and molecular dialogue inside the plant between light and gravity. And so in the absence of gravity, the theory is, well, maybe the plants will try and use light more or less. The role of the other tropisms, or the dominance of the different tropisms that control the orientation of the plant growth, starts to shift. And that was a theory, a hypothesis that's been floating. But yet the data to kind of support it is hard to find. And yet here it is in this database, just floating there, like a clue hinting at potentially that gene is the key to proving or disproving that theory.
Now, is this linked to the red pigmentation on the hypocaudal hook?
The red, kind of, yeah. How best to put it? So the red pigment, I'm not too sure about, but the hypocaudal hook is one of the ways we investigate this pathway. If you grow a plant in darkness, like the recent EXO launch, you see the etiolation of the hypocaudal as the shoot reaches. The hypocaudal is then hypersensitive to light. It's searching for it. And so, and our first studies, in fact, the study that we were looking at in this one, the brick 37 and the other one here, all the brick studies are basically done in darkness. So all these plants are up there in microgravity, stretching, lurking for light. It's just, the question is, does the lack of gravity have an effect on its capacity to search for light? And based on the genetic data that we've just stumbled across here, I would say, yeah, it probably does. Do you want to go one better? Oh, you do, don't you? Okay. So, so if only we
Don't
had
mind us, folks. We could be at this all day.
right. So if only we'd had somebody. I haven't installed the data yet. If only we'd had somebody who had grown plants in a 1G centrifuge, and then switched on a light. We do. Hi, Raoul. Raoul's a friend of mine, and so is Josh. And Alicia has just left our lab. She came over to visit. She's in their lab. She's a PhD student, but the coronavirus meant she had to return to Spain. Very sad. But we've now gone over to look at their study. So this is how they did it. They had a centrifuge spinning, and they had different samples along the centrifuge rotator. And so you get different fractional G-forces. And so if our theory about the perception of light, capacity of light perception to change in a microgravity environment is true, you might expect to see the light receptor FOT1 doing something interesting in this study too. Does that kind of make sense? So I didn't explain it that well because it's kind of complicated. But the moral of the story is now we're in here fishing for that next gene pattern. Standing Nation.
Again, here we're looking at hypergravity, not hypogravity. We're plus G's, yes?
Oh no. So this is microgravity. So there is a centrifuge spinning on orbit.
Okay, gotcha.
And right on the edge of the centrifuge, it's spinning at a speed that provides 1G. And there's photography in their paper, which you can get to by clicking on that and then going to Gene Lab, and there's one more click and there's the paper. So so they're spinning the sample, and then they turn off the centrifuge. So it's like switching off gravity. Kind of makes sense. It takes about half an hour to slow down. So unfortunate periods of intermediate gravity. Right. And then they switch on a light. The first time they did this study, they expected the roots to bend away. First time they did this study, the roots bent towards the light. Plant roots grow away from light. Negative phototropism. It's ingrained in them evolutionary from a long, long time ago. You can do some genetics. You can put some mutations in there, and you can create the opposite. You can make positive phototropism or negative geotropism. But like that result was really, really weird. Okay.
Could it be like, when we're kids and you get spin around real quick, you just get dizzy, you don't know where you are and you fall down? Can the genes just be doing that?
Maybe. But you could also be a pessimist, and you could assume that it's the first time that type of study was ever done in orbit, and maybe there were other factors that were not necessarily measured. Like oxygen disappearing around the plants as they are no longer being spun and they start to use up the local oxygen supply. And maybe you chose a particular variety because there's a natural variation in the world. And maybe you chose a very rare German variety that happens to skew and bend its roots in that direction when it goes into low oxygen. And perhaps it was just a coincidence that the light was right there. And so it looks like positive phototropism. The paper talks about phototropism. They go to repeat the experiment a few years later. That's the one where this data here came from. And this time they use a different variety. They don't use the weird German variety. They use the normal American Columbia wild type variety. This time they're growing the plants in the centrifuge with gravity. They turn off the centrifuge, so gravity disappears, and they start to use up the oxygen. And they turn on the blue light source. And this time the root bends away from the light source. It fits the hypothesis you'd have based on all the science, on all the history on Earth. So the moral of the story is when you go through old literature in the spaceflight environment, in the spaceflight area, you do have to be cautious because the early pioneers in this area are trying to design an experiment to fit in a tiny box that they're going to put on a rocket that they may never see again.
First-hand experience, right?
So you have to be forgiving, and the interpretation that they got from the initial experiment makes sense. But when you have enough extra flights and you have a few more studies that were done later, suddenly you can revisit and recontextualize the early data. Like, we're only human. But the moral of the story is when they did the study again, they did it with Colombia and the patterns look really interesting. And so now this would be a place where I would go in and I'd start fishing to look for what is the blue light receptor doing. Problem is, I don't have time to do that because this, as you can see here, in this study, in this app, we've created and linked together all these different studies. There are 5, 10, 15, 20, 25, 30, 35, 40, 45, 50. There's about 50 or 60 studies that have been plugged in. And I've got another like 50 more to plug in before I get those bloody mice. I was complaining about the GeneLab's mouse data. There's so much. Oh my God. Anyway, so
recommended quad core to run. Right? Yeah, what kind of machine do you run to actually process your data? Like, like you're just doing the visualization at the office or whatever.
Initially, I would use something called Cyverse. Cyverse is a free cloud computing infrastructure. Any teacher or researcher can have access to it. So you can have limitless cloud storage. So if you're getting loads of photos that you're accumulating on your local hard drive, you can stick them in Cyverse. That's how I share my time-lapse photography because bloat is an issue. Right? So yeah, but teachers can just save their images in Cyverse. Cyverse is great because it's linked to the NSF cloud computing infrastructure, which allows for cloud computing. So if you have image analysis software that you like, and you know how to use Docker, if you know how to use Docker, let me know and I can help you plug software into it. I'm still trying to learn how to use these Dockers and these other coding things. It's not my area, but I've got some image analysis software that I plan to put into Cyverse. So if you have imaging data sets that you want to put in there and you want to help me get the software installed, we can talk. Anyway,
that sounds good. We'll wrangle some folks to help us with that. That sounds fun.
It's a so Cyverse is awesome, free cloud computing infrastructure. But eventually what I ended up doing was working with NASA, and I basically become an advisor to the GeneLab program. So initially when we get our first spaceflight data back, we process it ourselves, and then we give it to GeneLab and they stick it in their repository. And then over time, they've built a collaborative workspace and basically process the data themselves. So data repository. Obviously, I was showing you my way of visualizing the data first because I think it's better, but I would because I made it, right? So you got to have confidence in your own stuff, right? But they have done a fantastic job of processing a lot of these studies. They've got about 250 in total. And so far, about 80 of them have been processed to a level where they can be used. And so essentially now what I'm doing, I'm just pulling in their data and using it direct. Because, and this was that study I was mentioning earlier, those guys, this was the one we were talking about, spinning plants in centrifuge. And so the data comes in this raw format. These are these FASTQ files. So this is the raw sequencing data, and they're huge. This is why when you talk about processing the data, I was like, instantly started talking about cloud computing, because when you've got like hundreds of these, what we call FASTQ files, these raw sequencing files, it gets big. I mean, like really big. So, so these days, after I process my own stuff using Cyverse, and again, if you want some tutorials on how to use that, I've got a load of stuff, but they've got tons too. They're awesome. But now, whenever possible, I try to just use the data that's been processed by GeneLab, because but having said that, this GeneLab repository, in my opinion, is not aimed at education. It's aimed at researchers like me and my professor. And what I've been able to do is build a tool that pulls the bits of relevant and useful data in here and make it accessible for biologists and teachers, in my opinion. And this is why, again, I've relished this opportunity to come and chat and introduce you guys to the website I've been building for Dr. Simon Gilroy as part of this NASA GeneLab program. We have an extensive education outreach program, and so the more people that come visit the site and use these data, the better, in my opinion. There's some. Do you want to see a funny video of the wizard? This is him, Simon Gilroy, on Japanese television, doing some outreach out there. He's hilarious. So as I'm rambling, I'm going to say what I'm going to do. I'm going to go over to a video that was made that promotes our work by the University of Wisconsin because it's the right thing to do. 4 minutes. Sorry, guys.
2, 1, engine ignited. And we have liftoff of SpaceX Falcon 9 rocket and Dragon. It's the biggest firework you've ever seen. Absolutely fantastic. Delivers new science and technology to the International Space Station. The absolute extra bit of it is we know in the Dragon capsule sitting on the top of it is like our experiment. First stage propulsion performing nominally. My name is Simon Gura, and I'm a professor of botany at the UW-Madison Botany Department. Research questions that we're interested in are all related to how plants deal with environmental stress. One of the practical aspects of the experimentation is to try and work out what happens to plants in space with the long-term goal of being able to use them as part of a life support system for the astronauts. It takes a lot of people to do that, and there's the Madison people and there's NASA people. Putting just one seed into space is a big deal. We actually ended up putting 1,200 seeds into space. They grew for a week, they were chemically stopped, and then it came back down on the same Dragon capsule that took it up after about a month. We got our samples back. Another one of the research team, Wang Yu, has been going through and beginning to do the extractions. Take the seedlings, we then do some chemical extractions, which allow us to concentrate and purify the RNA. We will send those samples off to the Biotech Center here, and the Biotech Center will run a technique called RNA-seq on it, which will tell us the level of every gene in the entire genome. You may think that putting this thing into space and getting it back and extracting it and waiting 2 years to get rid of it, that's the hard part. No, the hard part now is going into this pattern-rich data and saying, well, what went on? Can we see the pattern of low oxygen in these samples? Can we see the pattern of touch? So that is years of analysis that will come after we get that data back.
So we have bigger plants, they're grown in light. What other opportunities does this new experiment have?
We've been continuing our space research, so we've got some insights into how plants appear to be operating in space. The more we understand about how plants operate in space, the more easy it's going to be to design the hardware and the way that you cultivate the plants to let you actually grow them reliably in space. Very recently, that NASA program managed to put lettuce up on the International Space Station. That's awesome. Good. It's good. And so this has been grown on the space station, and they get to eat it. And so, of course, in parallel at UW, we grow exactly the same plants. And at the point where they're eating them, we pull out our plants, and in solidarity with the astronauts, we're eating the same lettuce on it. It was awesome.
So station R2 for veggie, the side of the pillows work without any problem. That's great to hear.
Getting to the point of having a relatively simple piece of equipment where you can grow plants and they get to the size where you can harvest them, that's an enormous step forward. Our next experiment is to test the idea that in space, because liquids flow in a different way and gases move in a different way than on Earth, that plants are suffering effectively a flooding stress. It's as if they're growing in a field that's been flooded, even though you haven't flooded them. What we need to be able to be doing is doing research to address things like flooding stress, or things like radiation stress, things like that. Take yourself mentally back to when people first started doing agricultural cultivation of crops. And they were trying to work out how to water them, where to plant them, how to select varieties. And we're thousands of years down that road. And so agriculture is incredibly efficient on Earth. And we're right at the dawn of doing space agriculture. It's a really, really unique opportunity for us.
Not bad.
Really fabulous. What a great intro. We'll add a link to that, just that one video as well, to get people to capture in. This all should be recorded on the Facebook platform, so anyone can watch and review it again. it's quite a long engagement. We weren't expecting to have Dr. Barker for such a long afternoon. It's so amazing. Come back tomorrow, would you? Come back, stay longer, right? It's like, I've got a few questions for you. And if we've got folks on the Facebook feed, if you've got questions, type them out there, and we'll see if we can't spend a few more minutes with Dr. Richard Barker there from Gilroy Lab and answering them. So with all this data, I have to ask you, how does machine learning and artificial intelligence play a role in the work that you do?
That is a good question. So at the moment, we're just starting to train a couple of simple AI bots to try and detect the light versus dark. But there was a moment a few years back when we had a goal. We could either choose to train the AI in-house or choose to make the data available. And that was kind of an option that Simon Gilroy gave me when a couple of years ago when we were like writing a proposal to NASA, and he's like, which way do you want to go about doing it? In-house with the AI or focus on accessibility first? And we chose accessibility first. I think
Thank you for that, by the way.
Like, I think I mentioned earlier, so I personally believe once we finish training the AI, it's going to point at the same basic pattern that we found through doing our basic statistics, which fishes out like the main kind of pattern. And then we're not going to understand how we went about getting there. Whereas I figured if we make all of the spaceflight data easily accessible and make sure that high school kids can access it with a teacher, like talking them through it, then think how many supercomputers, how much actual real intelligence would then be trying to answer the same question. I think a lot more. Time will tell. So yeah, so again, we're just now having to still have to do that because that's a very hot topic at the moment. And so we are starting to develop some code in that area. But the main difficulty is to train AI machine learning, you got to have big data sets. I mean, actual big data sets, like
super big.
Yeah, in the data sets we're looking at, so there's 40,000 genes in one Arabidopsis study, but like that's one rep. If you're going to train machine learning, you got to have like a lot of like replication. If you look at the intrinsic diversity in biological samples, so I saw a talk from the head of the twins project from Stanford.
And this is Mark and Scott Kelly, astronaut twins, right?
Yeah. And so n equals 1. And it was really fascinating to see that study. It's beautiful work done by some amazing scientists. But the guy who was in charge of it basically came out and showed this scatter plot. And I've shown you lots of scatter plots. And he basically said every single one of these dots is a different person. And if you monitor enough different factors, enough different like phenotypes, essentially everybody is unique and everybody is different. And so when you go and look at all the history of all the spaceflight studies too, I showed you that microbial study too. I showed you 2 identical microbial studies, and you could see them separating out into, so that even that same identical study wasn't like identical. So this is why it's going to be difficult to train machine learning when there's not enough data, in my opinion. And this is why I believe deductive reasoning and the and the child labor is the way.
Well, there you go. Well, that's a really elegant answer. Now, clearly, statistics is a big part of what you do, and your love of biology is evident. So, what did you go through in school? What, with respect to statistics and numbers and what have you? Where did you grab onto? What was it for you that really made it a spark to say you can now take these tools and apply them to your love? Where was that for you?
Well, for me, like this, when I was a youth back in the day, when the knights were riding their horses, statistics was not presented in an important or interesting way. Over here, like the way it is done in America, is all about calculus. And yet, I never look at the newspaper and see calculus misrepresented and being used to fool millions. So I think statistics is really important day to day, and I think we need to be more data literate in the area of statistics. I need to appreciate it more in general. The moment it really became important to me was when I was doing my first big experiment as an undergraduate. And there was a statistician in the department. The institute I was based at was where there's a guy called Fisher. He was a statistician who invented a lot of statistics back in the day. And so Fisher, like t-test, I think, on. And he, so we had a really good statistics department and a really good statistics consultant who came, and he came to me. I emailed him, I'm doing this experiment. I need some stats help. And he came to me and he looked a bit worried. And he's like, Richard, have you already done the experiment? And I was like, no, I'm consulting before. And he's like, phew, literally wiped real sweat from his brow. And I was like, what? He's like, well, if you'd already done the experiment, I could have only performed an autopsy and tell you how you should have done it different. But like, so I appreciated having a good statistician who would make a joke about what would happen if I did it wrong, and then so put me at ease, got me laughing, and then we sat down and worked out how to design a real experiment. And experimental design isn't that hard, you know. You got to have more than 4 reps, and you got to like assume the worst, account for a stochastic event. Something random is going to happen and something random is going to go wrong. So make your sample size bigger. So that way you can throw out the outliers. That's fine. If you can justify it, if there's a logical reason, if, not just doing it because you feel like it, that's part of science. So like people go, oh, I have to overplan things and make sure everything works perfectly. No, do it quick, do it rough, do it big the first time and be prepared to do it again. And that's how you're going to get past your fear of failing in science and actually start to create some data that you start to trust. Even if you do it right perfectly first time, if you're a good scientist, you won't trust that data, right? So experiencing the science, experiencing science methodology is how I really started to appreciate statistics. And then when I realized it's not a scary thing where you have to sit by yourself with a big book trying to find the right equation. These days, it's all in R and all in Python. It's 1 or 2 sentences away. And for a dyslexic person to say that, it's a big thing because even a dyslexic person can get the code right if they've got a good friend and they can reach out. I often mention throughout this talk about how my greatest strength is I'm willing to ask for help from people. And as you go through that and you get help from people, you start to work out what works and what works easy and quick and reliably. And I've collected those tools and put them here on this website and also on my Pinterest site, which is another tangent for teachers.
So you're going to share a link with that with us so we can make sure they know about your Pinterest site.
Yeah, by all means. And so on our Gilroy Lab homepage, there is this astrobiology course at the bottom. And so I just clicked on that and came here. There's a couple of terrible questionnaires. I don't know if either of these things work, but below again, I've got another one of these apps that I've made. And this thing here is again designed to introduce you to some introductory statistics and introductory modeling and all that kind of stuff.
That's a gold mine right there, for sure.
Also, this Newt's Voyage, it's a link to a game you can download where you go fly a spaceship around on different planets of different G-force levels. It's one of those like, it's a time sink and not recommended.
Is that like Lunar Lander? What were I can't remember that one game.
Yeah, it's exactly like that.
Okay.
So one of the things I've learned through the time is diving straight into statistics is a great way to switch everybody off. So like, so what's the reasoning for doing it? And it is this interplanetary transport system. Like, and I'm sure you guys have seen these videos.
Mm-hmm.
So I won't play them again because you've seen these ones before.
They're quite inspirational. You can kind of play them in a loop.
Yeah, right. So it's these things like the reason we have like the Falcon Heavy taking off, the reason we have now the designs of the interplanetary transport system is because some people did some math. So I'm sorry guys, I'm a biologist, I don't want to do math either, but we're gonna have to do it or we're not gonna be able to watch rockets take off and land. Have you seen the rockets take off and land? I'll tell you through the story and then we'll do the statistics. Mike's heard this one before from me, but like I think he'll forgive me telling it again, right?
9.
So this was the first SpaceX launch I got to see live, and we were like standing like just here. And my boss lady, she was watching it on her phone, and they got the live stream, and I started hitting it out of her hand because it was like, no, it's taking off. The live stream is delayed a few seconds. And then the rocket takes off and it's slow and it's ominous. It's not like the 4th of July. It lifts slowly. And you can see the sound wave coming at you. I had a physicist go, no, you can't see sound. No, you can. Like the birds flutter away, the water splashes. You then see the mangrove shake. And then it comes through the ground and up through the bottom of your heels, then up through the back of your ears. And then suddenly the bass is gone and you can't hear anything. And you're looking up and you're seeing like this rocket take off. It's awesome. And you look around and people act weird. I saw my boss lady and Simon kiss. Ugh, yuck. I saw these two American dudes give each other like high fives. These two like Japanese gentlemen bowing as the experiment lifted off to the heavens.
Wow.
And to my left, there was this one Italian, quiet Italian person. You always got to watch out for the quiet ones, right? Just standing there silent. And then he goes, look, there it is. And that was the moment when the SpaceX rocket had reignited and turned around in the sky. And then it's just like a streak across the sky, like a bit like Superman movies. The streak going across the sky, like without the blue and the red. And then you start to see it curve, and there's another burn in the sky. I'm going to skip in a little bit. And then just there it is. It looked just like that. And as it comes down, there's a moment just before it goes into the tree line where it kind of hovers above the ground, and then boom, boom, the two sonic booms hit you. And they are awesome. Like I didn't get goose pimples this time. I was sincere last time when I said I got them. and then it lands. And then it's real. There's this like silence, and you go home, and then you get excited for the data that's about to come down from that extraterrestrial environment. And you realize that when that data comes down, it's your duty to make the most of it, to make sure that for all those people invested all those time making the space station and making those rockets, that when that data comes down, that we're going to make the most of it. And so then you start thinking about that data in context of where we've come from, our Mother Earth, about how it makes us think, because the most important thing that ever came from space is that photo of looking back down at Earth and seeing our blue origins and just how fragile our delicate planet is. But it's not going to stop us, because Earth is the cradle of humanity, but we have to leave the cradle. We have to go and explore our new horizons. And when we get there, we need to make sure that we're planned to take the right things with us. And if we're going to go to the moon, when we go to Mars, we're going to need to take plants with us. And so can we model how those plants will exist in those extraterrestrial environments? Can we start to make predictions based on the research that we've been doing with Arabidopsis on the space station? So the reason we use Arabidopsis is its root system architecture is relatively simple. And so it allows us to start to do like mathematical modeling on like how water would move through it given time. But Arabidopsis is just one plant. It's in the Brassica family. And so once we start to learn like the rules about how Arabidopsis works, we can start to apply them to other species. And then, so over time, we can start to change species and the characteristics. And so this is like a very simple form of modeling, and we can start to appreciate the different types of root system structure. And now we have to bring into play the concept of gravity. Oh yeah, of course. Tropisms, one or two. So one of these tropisms, I forget which one it is the gravity feature. So we can you can start to play about with the sensitivity of the different root systems to the different tropisms. Like, oh, look at those roots going straight up. Oh, but I was doing so I've removed the gravity tropism component off the first order laterals, and you can see they've gone up here. Okay, cool. And
so help me understand to make sure I'm looking at it correctly. This is a recreation of the root development. And that on the left axis, is that an angle? Is it? That's depth in inches.
So yeah, so we have the depth going down, and we have the width. So we have, and then what we have in here are these different factors we can start to change. So sensitivity of the plant. And there's already some like mathematical modeling based on the different species, which is kind of described in this paper here. So you can select your like species of choice, like Zea mays, unleash Zea mays, like grasses have a distinctly different root system architecture relative to Brassica. So you can see the difference between maize and then say my favorite, or Brassica or Arabidopsis. And then one can come in and start to play with the different factors here. But anyway, so this is just one very simple modeling tool. And this is all written in R, but this is an RStudio app which has been put here to make it accessible. So ArchieDart is a software tool that's designed to pull data into these modeling environments. So I'm just going to run this one. This is actually the software that I used to measure our last spaceflight study. And so you can see we can measure growth over time, and we can start to look at the structure. So how would these structures change without gravity? Like, would the roots be trying to be shallow? Would they be trying to go deep? Would they go faster? Well, actually, the answer is shallow, but who cares? And so these are kind of like, so, and what do you measure? So you can like measure one feature, like the total root length or the total number of lateral roots, or you can break down all the different numerical components of the root system architecture, and you can break them down into like their principal components to do statistics, or make multi-dimensional like spaces out of them. So it turns out a sparse root is very similar to a steep root. As the roots try and get deep, they end up becoming like quite sparse in their structure. So how would these characteristics, how would these features change without gravity? So here you can see these different types of root system structures that can form based on these characteristics. But what role does gravity play in that? And so that's why we've started to collect together as many plant imaging data sets of plant root system architecture to try and build a database of the actual microgravity data to go with this modeling set. This is another interesting modeling tool to look at water movement and water pressure. And one can start to like play with like the fluxes of water through the system. So all of these tools here, these are classic modeling tools that are incredibly important for the future of global agricultural yield. If we are to continue to breed plants that are really efficient in our changing agro-ecosystems, we need to make sure we have varieties of plants that can continue to uptake water and grow conveniently, even in times when water is more sparse, when there's drought, or when there's more water due to flooding. And so these modeling tools have always been here. They've been around now, these ones for about 4 years, but putting them into the context of spaceflight adds a certain je ne sais quoi. It makes it a fun time to revisit these old mathematical modeling tools and ask, what questions can they teach us now? Marshall, I think this one allows you to change the water. There it is. Yeah, the water potential. So now you've seen one where we can change the water moving through the plant, water potential moving through the plant, which you've seen the root system architecture, how we can model that. And this thing here then starts to allow us to model like the water potential of the environment around the plant and make changes based on that, quantify things. So these are interesting. I think it's nearly coming to the end of that. Yeah. And so then this takes us finally back to the genetics. So so this is my favorite experiment, growing a plant horizontally. Roots go down, they're starting to forage for water. They're searching for the greatest water potential, right? That's essentially why they're going down. But in the absence of that water gradient, they follow the gravity gradient, because the gravity vector, because that's generally the quickest way to find water. And that's the main thing that plants do. And if we can measure enough of them, we can start to look at natural diversity. So this plant, as we measured it as it grew, we quantified the tip angle, and over time we measured hundreds of different varieties, made averages of those, and we can see the natural variation in sensitivity to gravity, which is kind of cool. And so we've been putting these tools out there so people can have a play. And then once we know the natural variation in gravity sensitivity, we can look at the different chromosomes. So Arabidopsis has got 5 chromosomes. I alluded to them earlier. They're just here. And so, yeah, we can look and find the regions, the chromosomal regions, so we have genes that are involved in the gravitropic reorientation at the later time points, 6 hours, 8 hours, or at like the earlier time points, 2 hours. This is the significance score down here, the LOD score. And so there's not many genes there that are involved. Over here there is. So this is that final piece again. So now what we're trying to do is bring it all together, right? So we've been looking at the genetics data. We've been looking at the environmental data. And now we've been looking at some modeling tools. And what we're now saying is, if we use all these things, we can find new patterns that haven't been explored yet. I believe the genetics is the Wild West. That's the new area. And the key will be to mine the genetic data, going into Toast, finding the patterns that make sense there, and then asking yourself, is there a way that we can test those theories, those predictions that are coming out of the genetics using simple modeling tools, the simple modeling approach like this? And then more importantly, is there an experimental approach? Is there a way we can start to design experiments that can test the patterns that are coming out of the modeling and coming out of the spaceflight genetics, and essentially use these data and these new theories to contribute to the next round of experiments? Oh, if only we knew a program that was about to launch some more plants to the space station. So,
nice, right over the net, right back to us.
yeah. And I think I'm done. I think I did my share for the day.
Oh my gosh, 5, 10x, 100x.
Seminar. Good grief.
Amazing. Wow.
I think, you know, the important thing for teachers and students, this is what math is about. It's like looking for patterns and patterns that lead you to questions. And ultimately, yeah, you'll come about, you know, what is the equation, what is the mathematical model to generate these things? But using a product like this and finding novel patterns and just trying to explore where they're coming from and what the parameters are. And all that is science is totally accessible to kids.
Ooh. And then the final bit to add to that is then the storytelling. So now you can see this tool, we've put loads of data and things and related tools together in one location. What the student needs to say is like, I'm going to become a storyteller, and I'm going to go into these spaceflight data, and I'm going to find a story that's so cool, that's so interesting, that you Exolab people are going to want to fly it. You're going to want to fly it. That's the game. Like, it's storytelling using data. And hopefully I've removed some of the coding so you can get straight down to the biological data, because that's what Exolab is all about, isn't it?
That's right. Yeah.
Yes.
You've made the gold pans. Now we just got to go panning ourselves, right?
Now that's the goal. That's the thing. And so I think I shared with you the manuscript we wrote that described how we use it. It's everything that, to know how to use it is in that paper. So again, if you could circulate that, I would appreciate it.
Most definitely will. And yeah, we'll make sure there's access to it through this feed, through the link there on Facebook. And I also understand that there's a lot of folks that aren't on Facebook or prefer not to be. So we're going to be running a broadcast through the next 35 days with gentlemen and women and gentlemen like Dr. Barker here in different fields of study, both on YouTube and on Facebook. And what an extraordinary journey. I'm going to have to rewatch this a number of times and hopefully glean a little bit more. I probably grabbed about 5%. It's so rich and inspirational. You really kind of have provided the tools for us to kind of explore on our own. If we're on a different world and you basically give us a suit to protect us from the elements, right? So we can kind of go out there and kind of get right to our research as opposed to worrying about just surviving, which is a great asset. Michael, did you see any questions, any additional questions on the Facebook?
Let me just check.
And I think we probably...
Lori Waters says hello, Richard.
Oh, hi, Lori.
I did have a question, if there are any others out there, about in situ PCR. The ability that you're doing some kind of fixative probably, you're bringing it home, you've got that gravity vector, it takes a couple days, if not longer, to finally get it to the lab. You then have to go to the process. And I know there's like Genes in Space that's up there that's doing some PCR work. Are you guys doing anything in situ right now?
Not right now, no. So far, we use concentrated ammonium sulfate. It's an RNA later chemical. It fixes the nucleic acids. It's basically a super concentrated salt solution that makes proteins precipitate out of solution. So it denatures the enzymes that would degrade the nucleic acids, the DNA and the RNA. And so that way we can then, so it's chemically fixing the plants, so they can be frozen to -80 and then returned to the lab for RNA extraction. I know of PCR programs that have been done on orbit. So far to date, they've always been proof of principle, as opposed to here is a tool that an average researcher can use. The other thing is a PCR is relatively laborious. to set up. It takes a lot of astronaut time. So when designing spaceflight like experiments, the goal is generally try to make them astronaut proof, as we like to say, which is keep it simple. Like you don't know how much they do so much. They're amazing people, don't get me wrong, but the simpler the experiment, the less likely it is that something can go wrong.
Right. They touch our payload twice.
Yeah, and that is a privilege too, right?
Yeah.
It really is. Like,
oh my goodness, we're so grateful.
I have a question, if you don't mind.
Yeah, go
right ahead, man.
Yeah. So anyway, it's just so much I've gotten from what you've been sharing, Richard. One of the things that struck me was just how you were impacted by the process, and that included, being someone who's involved in looking at the launch, doing all of this development, and in particular, you showed some videos where it was on station and it looked like astronauts are working with your experiment. And I know sometimes they do that by themselves, but many times researchers such as yourselves are on the ground looking at the 6-pack of video feeds coming down, and one of them will be the astronaut. So I'm curious, can you share anything about what it's like being that kind of a researcher where you are literally working in real time with an astronaut on your experiment? And I don't know whether I'm reaching too far with that question or whether it's something that really resonates with you, but as to whether you've had those experiences.
Yeah, no, that's a good one. I'm just trying to find like the video of there's a video to go of everything, obviously.
Yeah, of course. This is such a rich data set.
So hopefully this has got the right one on it. So there was that moment after the launch, it just taken off. The bosses had kissed, all that stuff, right? And then you hopped on the flight, and you go home, and there's a moment where it's a bit of an anticlimax, a bit of a dip after the rush, and then like you get a phone call, and it's like, quick, right, everyone back to the lab. Okay, so let's go back to it. Everyone quick back to the lab. So SpaceX 13 had docked. We were following it on Twitter. We'd seen the launch, following it on Twitter, and it didn't miss. Woo-hoo, right? And we take it for granted, but recently with the recent, was it? I won't speak. Yeah, Boeing's recent miss. Thanks for that. Yeah, so it's rocket science. But anyway, so it docked. We got excited, went back to lab, and we got this live stream. You can even see, like, because this is just screenshot after screenshot. So this is our own time-lapse photography. So most people think that astronauts just hang out on the side of the space station taking awesome selfies like that, and occasionally they get to that, but they do loads of experiments. At any moment, there's like 250 experiments ongoing on the ISS, give or take. And this was the lucky moment we had when we handed over our samples. These petri dishes are the ones we'd sown the seeds on ourselves, and you see him going in there. And if we play it again, look for the moment he gives us the thumbs up, because so now we're really strict on the protocols that you do. Everything has the stickers. You have to approve all the stickers they put on there. We went through 3 different types of stickers to get, there was the thumbs up, right? We wrote a secret message on the side of one of the tinfoils where we said, "Thank you, Scott."
Oh. And he saw that message and then gave the thumbs up. Oh, so this is Scott Kelly?
This was Scott Tingle.
Scott Tingle. Yeah. Okay.
Yeah. We did get to train Scott Kelly, but we were delayed to launch. So we didn't get up there when he was there.
So were you watching this? I know sometimes you have the privilege of watching, of actually being on the ground in communication with the astronaut.
Yeah, so we got live streamed to our computer in the lab, but we then had to talk to people at Johnson Space Center, who then relayed the message up.
Oh, okay, got it. That makes sense. Protocol.
Yeah. Since then, the Wizard and my boss lady Sarah have both received the next level of training to be in that next room, and it's hilarious. So there's 20 people in the room. They're all regulars, and they all have a different acronym for their role. It's all code name. You're not allowed in the room unless you can reel off the code names for every single person in that room. And yeah, it's hilarious. And so they came back after the training and they're trying to remember all the opnoms for different people. And it's just, mind-boggling.
Thank you so much for sharing that additional insight into what it's like, literally, when you are watching someone live working with your experiment, your precious experiment, and you're in that communication chain actually while it's going on. I think that's one of the remarkable things that being involved in this space world sort of really provides access to. It sort of it really does get you connected in real time with what's going on the space station. So thanks again.
It's those subtle details. My pleasure. It's those subtle details you don't really notice. So you see he's holding up every plate, and that's so we can read the letters and the initials that are on there. And we have down here on Earth, we have a diagram. So every plate has to be in a specific position, and it has to be in a specific orientation.
Wow.
So as he's doing that, we were leaning over going, did he do it the right way around? Kind of checking these things. And there's a couple of moments he had to go back to double check they got them in the right place and the right orientation. Because those petri dishes have a top, they have a bottom, they have a front, and they have a back, and they all have to line up. It's the subtle details. You have a little thing I'd like to point out. You see that little star there, the little zinnia flowers?
Oh, yeah.
That's a tribute from the American Society for Gravitational Space Research. If one of their founding members passed away a couple of years ago, and they had this plaque put there in her honor. So it's just these subtle little.
Superb.
And then this is what it all comes back down. He puts the plants in this glorified petri dish and they come crashing back down to Earth. But then you've seen all the genetics that comes after that. So the next 300 slides of genetics won't be
300. Some might think that you're exaggerating.
On this,
but no.
But no, really. It's like already. Oh god, this one's got smooth.
Yeah, it really is. Look at that. Yeah,
65.
Just shy. Yeah, there we go.
So I was exaggerating.
Round up.
Ooh, but actually, okay. So this is all high-end stuff. I'm not just bragging. I'm really excited by these days, and I think they're really cool. But everyone can get involved. There's this wonderful piece of software called Easy Leaf Area, and I actually used it to analyze some of the data, the studies, the plants that came back. All you have to do is take a photo of some plants in a known area and put a red square next to them. So all of your students in your classes can grow some plants to a certain age, put a red square of known size and take a photo. Easy Leaf Area, it's free. It can run on a Windows operating system, or there's an Android app that you can download to your Android phone. And essentially it calculates the green area in your given area. So this was the data that came out. And we can see that our wild type plants had a dwarfing effect. Their leaves were smaller as a result of the spaceflight environment. And the varieties that we knew were resistant to flooding, that are actually, their leaves are the same size in the spaceflight environment. Well, almost. It was interesting that a tool is so simple and easy to use, like this Easy Leaf Area, can produce data which is truly, really insightful. So again, moving forwards with your classes, I urge you to see if you can get some students to grow some of the NASA target crops and some Arabidopsis or some other related plants and practice using this tool, because I think it has a lot of potential for collaborations in the future.
So, Dr. Barker, our first 3 flights were with Arabidopsis, and we kind of went into microgreens, and now we're with the legumes. In terms of just the general research in botany and what have you, would you suggest that we consider, as we move into 2021, going back to something like Arabidopsis as a kind of foundational research plant?
In my personal opinion, my advice would be, because you guys have done all these awesome launches, the biggest problem about spaceflight biology in the moment is the lack of replication. And what I mean by that is the same study that's gone up again. When I look through all of these NASA GeneLab studies, the thing that I've noticed is majority of them have not got imaging that goes with it. There's loads of plants that have gone up and they've just, or cell cultures, plant cell cultures, they've gone up and they've just come back, brought the tissues down in RNA later, and then just done RNA-seq or microarray. And I think what some of the things I was trying to show you is if we get photos of the plants, there's a lot more wealthy data there. And if we can correlate the imaging to the genetics, it just adds so much more value.
How about even broadening that spectra from visible light, say infrared or ultraviolet?
Exactly that. You could do infrared photography of them. So I would be tempted to try and look to some of these early data sets. And again, if you want to go offline, we can talk about them. I'd point out my faves and that. And just replicating the ones that where we have the genetics, but don't have the imaging to see if there's patterns that we can link up there.
I'll have to share a picture and dig it up. One of our early prototypes, it just wasn't practical in the volume that we had, but we had literally a turnstile camera that was basically taking images in a 360 as the plant grew. The idea would basically, so you could rotate it in 3 dimensions and see the entire plant over time. But the prototype, we couldn't get it to production at a reasonable cost. And there's a lot more work on that.
I've tried to do that myself a few times with like the rotating camera. And it's hard, isn't it?
It is. Yeah, more failure points, right?
Yeah.
But as you know, Richard, as right now, we can provide you hour by hour images.
That's the most important thing, really. You don't need anything more than an hour. But yeah, I would love to see these data that came back off your previous ones, and to see if I can push them through the same pipelines that I've used in the past.
All right.
What would be great is making recommendations on what we could do to be much closer to where you are on the research front. Kind of tail wagging the dog, but I think we get teachers and students excited alike. And as we increase our fidelity through insights that you provide and others, I think we're going to have something that's really quite meaningful for this next generation.
I'm also really excited by the microgreen program that you mentioned. I'm just starting a new collaboration in this year. Obviously, the coronavirus has delayed it. With the Kennedy Space Center, my friend Christina Johnson. You could probably get her on here. She loves talking about spaceflight biology. She's a great educator. Yeah. She's provided the protocols that they use at Kennedy Space Center. And so I was meant to be doing it this summer with the UW People program, but that might be canceled now because of the coronavirus. But our goal is to do a new round of microgreen research in a way which is compatible with my genetic methodology, so we can use so we can link it to the Arabidopsis genetics, and then also trying to because I know that Christina's going to fly some, and I've got a microgravity simulator, a clinostat, which I'll be going them in. Ooh. And I've just been told by my loving wife that dinner's nearly ready.
We're late for dinner.
Well, way beyond the Call of Duty, Richard. Yes. We're indebted. Anything that we can do, including staying out of your way. Your research is I'm sure very important. We'll try to keep a very high signal to noise ratio when we reach out and think about possible collaborations. But we're very excited, and you're super inspirational. And I kind of at a loss of words at a certain point. It's really exciting to hear what you have to say.
And so, I'd just like to finish by saying thanks for giving me the opportunity to share the research that we do. Please share these links with teachers. If any of them create interesting lesson plans, I would love to get them back just so I can recirculate them. That's my goal. Again, I would love to see those images that you have from your previous Arabidopsis and microgreen launches. I'd love to see if any of my image analysis tools would work on them. I would encourage you to get students to try playing with that Easy Leaf area software. It could be a good way to create a citizen science project, which is similar to the Growing Beyond Earth program. And we'll be planning to do that with the microgreens in the future. So I hope we can collaborate in that area too.
Oh, that introduction, by the way, would be fantastic with respect to microgreens. And if she wanted to join us later on in the 35-day program here, by all means, we welcome her.
Richard, is there anything on akin to Easy Leaf that's compatible on OS X or iOS or Chrome operating systems? Those are Chrome is become really big in schools.
I see. I know that's what I've been thinking about too. I'm still in the process of trying to find the optimum way of deploying software. This was why I mentioned earlier the Cyverse. Cyverse and their Docker system allows you to build like a environment for you to install their software tools. I believe this is will be the best way to do it in the long run. It's a set of code I haven't conquered yet. Again, if you know anyone who's familiar with Dockers and just building like computational pipelines, I'm again always open to collaborations. Because the key is to have the image analysis software in the cloud, and to have it as automated as possible. And that's why
we'll scour our network. I'm sure we can find a few folks that would be excited about collaborating with you on the Docker project.
That would be awesome.
Super cool. Win-win.
If that's the way to do it. Right?
All right. Well, let's not get your wife angry at you. It was a pleasure that you joined us today. And for all those that are online, our sessions run through till 5 o'clock today Pacific. So we still have about 30 minutes in our program. Might even run a little later. As we all thank Dr.
Thanks,
Parker,
Richard. That was
very
amazing.
much, sir.
Thank you.
What we'll do is maybe take a 3-minute break, and we're going to come back and we're going to review the Leguminaut challenge and how your students, again, anywhere from kindergarten up to university, can be involved in selecting a candidate legume to fly in our mission on SpaceX 21. So why don't we give it, it's 4:30 on the hour here of West Coast. So let's give it 3 minutes or so. I'm going to step away, just a quick water break, but hang tight. We're going to come back and talk about Leguminauts. All right. That was fantastic, wasn't it?
Bye for now. Thank you.
All right. Take care.
Cheers.
So back in 3. That sound good?
You're still going to keep the stream running, right? You're just going to.
I'm just going to step away for a sec.
Yeah. Okay. No problem.
All right.
How you doing, Michael? You're always looking good.
Yeah.
When I grow up, I want to have a streaming background like you've got. It would help if I just switch it back to me. There you go. In comparison. Although right now I've also got this little puppy
there you go.
streamed in. So my little Lego ISS above sitting there doing its thing.
Yeah, and there's students out there that are building various NASA themed Lego models. I got one of my students, 4th grade girl, is building the Saturn V.
Oh yeah.
Haven't seen a completed photo yet, but she was doing a good job.
Of course. There you go. Back to the good old Earth. But there we go. Well, this has been an awesome sort of switch from, other current news.
Yeah.
This is perfect.
No doubt. It's an important distraction, but there's a lot of good stuff to work with here.
Oh yeah! Wow.
And our thin blue line.
Yeah.
All right, I'm going to take a quick pause myself.
We'll
be back in a couple.
I'll see you back in a minute.
Yep.
All right. Hello, Facebook. So we've had amazing multiple hours there with Dr. Richard Barker from the Gilroy Lab, taking a super deep dive in all the amazing world of microbes and plants, and really data science. As we quantify research, what can you discover? He gets down all the way into the mechanics, the molecular level of what's happening, the expressions of genes as they're affected through things like light, phototropism, or gravity, gravitropism. The ExoLab is a device that's much simpler than the deep level of research that he's doing. We hope through our missions that we're able to bring something back to Earth that is capable of doing some sequencing, some RNA sequencing on our experiments. The labs themselves are really designed for us to function and send multiple missions up per year. So our next mission is in August on SpaceX 21. That's mission 8. And it is the third mission in a series in which we're studying the symbiotic relationship between a bacterium, Rhizobia, and the legumes. And our first mission that was in the series was mission mission 6, IsoLab 6, and we were looking at the right bacteria that we'd want to use to study this fixing nitrogen. So we ran Azotobacter and Rhizobia and an E. coli control on little 35 millimeter plates. And we down selected to Rhizobia. We then selected cowpea, Vigna, as the plant we wanted to investigate in this relationship between the bacterium and the legume. The bacteria able to process the nitrogen and provide that to the plant through its photosynthesis, delivering some carbon. Really nice trade going on there, the plant and the bacteria forming this relationship. And there in the roots, these nodules will develop under successful growth. Cut that open, there's almost like a hemoglobin in there. You can see the pinkish red. In fact, Michael, you might even have some samples you can share with us on that.
Yeah, let me just look.
That was our flight candidate on this current mission that we're literally bagged up today. I'll have to dig up some images, but our flight provider, a big shout out to Space Tango in Kentucky. They're the ones that help us get to orbit every time, and they have an amazing crew and team, researchers and engineers that help us fulfill our mission of bringing learning right to the very edge of discovery. And Dr. Barker definitely took us on that journey earlier today. The mission that's coming up is Flight 8, ExoLab 8 for us on SpaceX 21, and we need your help. And we thought this would be a perfect opportunity. Many of you students are at home. You could be a parent watching this, or maybe you're a teacher thinking about what you could do with your students. They might be second graders or eighth graders, or they might be you might be a student at university just excited about astrobiology. And so we need to select a number. We have a number of candidates that Michael Wilkinson, our lead teacher, will walk us through in terms of just the orientation on our challenge. But we're going to down select and find our flight candidate in the next 35 days. Today's the first day of spring, so happy spring, everybody. But we're going to conclude this on Earth Day, April 22nd. It just happens to coincide this way, but it's going to give us just enough time for our principal investigators. We have teachers that are directly involved in our missions to help us write the lesson plans and experiences for the students. Many of our students are working inside states in the United States here based on the next generation science standards. So in addition to the disciplinary core ideas and the cross-cutting concepts, we're integrating the science and engineering practice in that as well. And so help us determine it. We want to have a little fun. And our youngest students, we welcome art, poetry. You want to do, as I think someone talked about, interpretive dance, go right ahead. We don't have hashtags yet. We'll have to figure that out. All this has kind of been done on the fly. There is no cost for any of these programs. Tony So, co-founder at Magnitude, will walk us through briefly on the platform to show you where to go and how you can register and set up. But from there, I'm going to kind of turn it over to Michael Wilkinson, and he's going to share with us our general challenge to you. And this broadcast is going to happen again tomorrow on Facebook, but we're also going to send it over to YouTube, and we're going to spend a couple hours over there as well. Dr. Barker won't join us. I'm not sure if we're going to have another VIP guest. We're getting the kind of calendar lined up as we speak. This was put together literally over the last week as this pandemic has kind of affected everyone, and the shelter in place is in order. Most schools have closed down. What can we do to kind of continue the curiosity that all of us have in kind of a focused way? Come join us, help us find a solution. We need a flight candidate to send to space. So Michael, I'll turn it over to you.
Okay. So you mentioned some of the plants with inodulation. I was just thinking maybe tomorrow, I think I've got some that we can uproot and actually do a little bit of nodule analysis online, and maybe even do a dissection. So we'll see if we can make that happen in the next day or two. We'll make sure that's on the calendar so people can plan for it. But here's the deal: we've got to survive in space, and as you heard Richard talking about, we're looking for sustainable means. So we're not in a situation where we can ship everything that we're going to need. Food and water being two of the heaviest and most difficult items to deal with because they're perishable. And if you've ever gone on a really long camping trip, you know that eating processed, dehydrated, irradiated food for very long can get really tiresome. And this is a legitimate issue with the astronauts. Is if your palate's not stimulated, you start to eat less and less of the things that you need, and getting that important balanced diet is critical. I think I just froze. There we go. I froze. I don't know how long. But so having that balanced diet is critical. Getting certain nutrients and not having some nutrients, we're finding, makes a big difference in terms of retaining skeletal density and muscle mass. So we're looking at high nutrient plants. The microgreens that we are that we did previously all had good nutrient values and taste involved. So that's where your students might come at this from the culinary standpoint. Which of these legumes do they find the most appealing to eat, or lead to some of the most interesting foods or the most flexibility? Any reason that you have for proposing the legume is valid, whether it's scientific or more humanistic, because we've got to deal with both of those areas. It's about really keeping humans alive and thriving, not just existing. And then of course there's applications that loop back to Earth itself. We've got a difficult environment on the space station. We've got a high carbon dioxide level. We have high temperatures. So it becomes a good analog to think about how critical food crops are going to be affected by climate change here on Earth. So of the legumes that we've proposed, we flew cowpeas. Also, it's the same thing as black-eyed peas, but it's a kind of a bigger grouping. Flew cowpeas this just concluded, and now we're looking at, do we fly cowpeas again, or do we fly another legume as our plant model to see how that symbiotic relationship between the bacteria and the plants function. And that's critical because we can't use regular fertilizers off Earth. It's either too expensive, whether it's energy or literal dollars, too heavy, or dangerous. And so we're looking for a biological solution, and that's what these microbacteria that live in the soil, the rhizobia, do, is they fix or convert about 80% of the nitrogen gas that's in our atmosphere into the nitrogen compounds that plants need. And the class of plants, legumes, they're not the only ones, but they're the most common and most abundant of species that actually can work with the bacteria. And it's actually a really interesting relationship in terms of our times. Viruses are a little bit different, but here you've got a bacterium that is pathogenic. It actually wants to invade and damage the cells of the plant. But there's a switch that flips in this relationship when everything's all correct, that actually changes that pathogenic organism into a symbiotic one. And so what the rhizobia has the ability to change the nitrogen, the inert nitrogen, into ammonia, and then into the nitrates and nitrites that the plant needs. And the plant in exchange is giving the bacteria some of the minerals that it's been able to metabolize from the soil, and most importantly, the carbon compounds, sugar, that the bacteria needs for energy. So it's one of those situations where both organisms are benefiting equally from the relationship. And Ted at times he's described it as it's like a barter system. I've got something that you need, you got something I need. Let's work out a deal. And over millions
put it in
of
simple
years,
terms.
over millions of years of evolution, this currency of nitrogen and carbon has turned out to be a pretty viable solution. So we're looking to see, can these nodules actually happen in microgravity? So that's kind of a quick overview of some of the science that we're engaged in.
Let's give them a little visual if we can. Our team at Magnitude, we're a small team, but we definitely punched in above our weight here, providing, I think, a real fun experience for all ages of students. Tony, can you maybe set up the landing page, share that, and can you walk us through what this looks like? I think there's a great visual also we've kind of lined up where we're looking at our different candidates. And there's more to come. We're adding more to this. If you have some interest in being more directly involved outside of just kind of on the edge, reach out to us. You can send something to team@Magnitude.io. It's going to get to our entire team, front end, back end, front office, back office. And
yeah, maybe you've got an experiment, or a student of yours has got an experiment that you've always wanted to be more than a one and done. Here's a chance to get some crowdsourcing and get lots of people involved with you.
Exactly. Hey, Tony, so this is brand new. So I don't know if I had a chance to see the released version from midday today. Why don't you walk us through this?
Sure. Hello, everyone. This is Tony So. Now, all you have to do is go to Magnitude.io website. And right across the top, you can click to learn more about the challenge we have just started with. And this is the landing page. It will give you a lot of information about the challenge. And also down here below, it has a schedule of all the video conferencing that we have set up, and we will have additional VIP guests, populating on this chart here as we have it.
Yes, and if you missed it, if you're just joining us now, getting this review, Dr. Richard Barker from the Gilroy Lab. Wow, did he take us on a journey? It's right. I believe it's all going to be recorded there on Facebook. So if you start it the first 4 hours, I think, wow, amazing. Let's turn this into a DVD. Look for more VIP guests coming over the next 35 days.
Right, and a little bit further down on this page, we have a number of candidates for our next experiment. And each one of them has a team lead, and each one of they are all PIs in this experiment, and they will be the lead for each candidate. So make your choice on which one you want to work on and discover if that is the right candidates for the next experiment.
Yeah, and if we could dig in a little deeper here and see, I don't know if you're online with us, but if you are, please jump in. We're creating stats for these legumes. Out of 100 grams, how much of that is protein? How much of that is carbohydrate? What is the strength or weakness of this particular legume? Is it resistant to temperature? Does it like a microgravity? Is there some signal in its DNA that's going to make it do a little better? In some prior study, if we can find one, maybe there's an argument for doing that. So we're not sure what we want to fly. We have some idea, but we want to put it out there to you as students, teachers, develop your hypothesis to bring your group together to say, we're all about alfalfa, we want soybean to be the lead. And here's our argument or reason why. Create your hypothesis, provide substantive information. And we're expecting that if you're an older student, and if you're in grade school, if you're working with students of younger age, they might just be getting into understanding their numbers and being able to read and write. Have them illustrate something. Have them share what they think. And as I think Michael said, let's get into the culinary. You might
we're
like
all,
a
we're
particular
all
candidate
relying
because
on
it
canned
tastes
vegetables these days. So it's, you know, make a run through the canned foods department and pick up or get a dry bag of them and do some cooking and tasting. And I mean, if you've ever done that with your students, you know that that's a lot of fun. And it's something that it's an easy way to insert all kinds of curriculum into that. You can have them writing stories, you can have them doing mindful eating and write some poetry or draw some pictures or tell a story or make a graph, or, you know, just there's so many directions you can go with it. And I know all the teachers watching, you know, I've been at this 3 decades, so it's like we're a creative lot, and it doesn't take a whole lot of an idea to, you know, flip the switch and get you thinking about your individual group. So, you know, that's one of the biggest things that, you know, kind of a personal goal here is we're not trying to prescribe that you do X, Y, Z through this social isolation, social whatever we're calling it, COVID-19 mess. But we do want to give you a sense of empowerment and you know, maybe, you know, a stimulation of something that rises above the noise that's out there, and we can collaborate on something together. And anything that your students contribute to us around this is valuable and valued. We really
have some fun,
want
right?
those. Yeah. I mean, you know, the PIs, we were all, the night that we all signed up on which of the legumes we were just going to take, we got silly and we all started thinking of each of them as superheroes. So, you know, the fun is important.
Right. So for those teachers who really want to be part of this experiment, and all they have to do is sign up to our platform. And right now it's entirely free. All you have to do is click on this button, and you will be taken to this one sign up page. You can sign up with your school account, whether it is from Google or Microsoft. All you have to do is select the button. And for example, if your email is from Google, the single sign-on with the Google will instantly authorize to have your name and your first name and last name and your email address populated here. And there are only really 3 steps. The first one, the second one is you know, take a look if your school district is already in our system, and if it's not, you just create your new school district. Let me go back to the other page. Hold on. So let's say I already have a school district in our system, so I selected it. And same thing, once you already have if you don't have a school set up in this one particular district, you can create a new school. And if not, you can just select the school that is already in the system. Now, you are done. All you have to do is click on create account. Now, the reason why you are seeing this is that I have already used this email address to create my account. Now, once you click this, it will instantly, okay, it didn't. Yep, it will ask you to log in using your regular email account, because space.magnitude.io, we have already created an account. So over here, this is your new dashboard. Now it will tell you, ask you to create your classroom. All you have to do is do that. And you set up, for example, this is going to be 8. challenge. Now you are inside your class, and all you have to do is give your student this class code and the sign up URL, and they can just create their own account under your classroom. Now, let me go back to the dashboard. Now we have set up a number of free lessons for this challenge. You can either click on that and it will take you directly right into all the free lessons under this challenge. Now with this, you can click to look at the entire lessons section by section down here. And if you really like this lesson and you want to assign it to your class, all you have to do is click on that and select the class that you want to assign this lessons to. Select the start day, for example, and then the end day. And then boom, that's all it takes to assign a lessons to your class. Now, for your students who sign up to your classroom, there's nothing for them to do until you assign them a lessons. So I would truly advise you to do that. Now, let me go back to the lessons. Now, if you really like this lessons and you found that some of the wordings are too advanced or too hard for some of your students, all you have to do is click on Chrome, and you can clone these lessons to however you want to lessons. So once the lessons is cloned, you go to my lesson and you select this lessons. Now you can modify every single section in this lessons to the way you want to teach your class. For example, if you want to have one setup for your 5th grade, 8th grade, and 9th grade, you can really tailor each one of them, tailor the lessons to each classes of yours.
Yeah, I had a group this spring or winter, I guess, that a significant number of the kids in that group had some reading issues, and so I actually went through one of the critical lessons that we were working with, and enlisted the help of a couple of them, and we revised all the text to make it more accessible, and the whole class had a lot more fun with it then, because they weren't struggling so much with the vocabulary. And likewise, on the other end, if you got kids who could really take something much more deeply, take it further, do some more statistical work with it or something like that, here's a great way to insert those pieces as well.
One thing I do want to point out is that we're going to have a number of opportunities to chat with you throughout the next 35 days as we prepare to identify the candidate that's going to fly on our ExoLab 8 on SpaceX 21 in August. And so right now it might feel a little overwhelming. We had a super deep dive in microbiology and astrobotany for a few hours earlier today. This program is very malleable. It's very flexible. You might have very unique pedagogical requirements. Your students may have equipment and internet access, they may not. We welcome your questions and challenges, concerns. We want to find ways to take amazing experiences. What we're doing here at Magnitude is only one example. Over the next 35 days, we're going to be showing you other really cool examples and neat things you can do aboard the International Space Station, 250 miles above our head, traveling at some 17,500 miles an hour, with real actual science research your students can participate in. So just keep that in mind. It might feel overwhelming right now. We're going to be popping back on again tomorrow on Facebook and on YouTube, and next week as we start to fill out our calendar over the next several weeks. So keep an eye on what we're doing here. It's take a little while to get oriented, but I think you're going to dive in with some really exciting and fun stuff.
And one more thing I wanted to add is that right now, when you click on the lessons library, all you see is 3 different selections. Pretty soon, we will have ISS Above lessons presented as a free product. Thank you for Liam Kennedy. He has decided to put his free lessons on our platform to make it available for students around the world to use and access and really learn about International Space Station. I can see that Liam is still on the line. Liam, you want to describe your lessons a little bit?
Yeah, just hold on a sec. All right, I'm back in the flesh here. Everyone hear me okay?
Yes.
Very good. So thanks very much for that opportunity. The lessons, just to give a shout out to the ISS National Lab, were created through funding provided by the ISS National Lab. So it's a set of, and you can access those lessons online on my website right now. But the advantage that I'm really taking from having them on Magnitude.io's platform is that it's something that can be much more deeply integrated into the way in which teachers conduct lessons and activities with their students. So I'm very much looking forward to having them on the Magnitude Classroom platform. And thanks again for making that available.
Of course. I think that's all I have for now.
Thank you, Tony. So that's Magnitude.io. The instructions will be pretty set up there on top, describing what you need to do. We're going to walk through a tutorial in more detail tomorrow. What was the start time? I think it was as early as 8 a.m. Pacific, 11 Eastern, for a open conversation around our legume challenge, selecting a legume to fly on our ExoLab 8 in August on SpaceX 21, as well as a little more background about what Liam is doing with ISS Above and things like orbital mechanics and what have you. There'll probably be some summary links and what have you as we digest all of the rich information that Dr. Barker shared with us earlier today. And we might even be able to tease what next week's schedule might be looking like. Again, look at us as a resource here. Everything is free and available to you. We're here to support you and your thinking and pedagogically how you're going to approach your students and their needs. Every community is different. I think every district has their own resources. They're varied across the board. We're looking at from analog to digital, from the youngest to the oldest. What can we do in that matrix to make it fun and engaging for you, for us to build community as educators and really have a great time in kind of a little difficult time as we think about taking care of our families and our loved ones. And then on top of that, having to think about others like our students and whatnot. We're here. We're excited that you're joining us and you've popped in and said hello. And please look for our, be friends of us there on our Magnitude page there on Facebook. And look for that for more information coming up for tomorrow and the next 35 days. So I guess we are just pretty much at the top of the hour. For our teachers that have been joining us on ExoLab 7, as you know, they put us back in the flight bag today, getting ready to send us home. I understand it's going to be April 10th before it is back on the ground in our hands. So we'll keep you abreast of that. Our standing call will be regular next Wednesday for all those that can make it. And so thank you for joining directly here in the Zoom hangout. For now, I guess we're going to wrap it up. We went right through lunch.
See you back 1500 tomorrow, UTC.
Wonderful. Thanks, guys. And be safe out there. Wash your hands. Take care. Bye.